Welcome to this fifth class of the protein engineering course based on artificial intelligence. And on this occasion, we are going to review the topic related to the structure of proteins, as well as all the molecular interactions that exist within them to obviously maintain these three-dimensional configurations. Remember that this is only the fifth class of a total of 10 that make up the course, which is completely free and can be found available on YouTube, so you can delve deeper into each of these topics. Likewise, before starting, the announcement that all the images, all the quotes, as well as recommended literature for each specific topic is in the notes section of each of the slides. So for more information about the course, please visit its official website. Likewise, I invite you to review the associated GitHub repository, where you will be able to find tools, databases, as well as other courses related to biology, bioinformatics, and data science in general. If you like to download the slides to access the information or to be able to adapt them to a course you are developing, you can do so from the Zenodo or Google Drive platforms. If you also like to leave a question, you can do it here in the YouTube video in the comments box section, or you can send it to me via direct message on one of my social networks, where you will find me as the user at Myinger. And finally, if you find the course productive for your professional development and you like to reward it financially, you can make a financial donation via PayPal in the following link. So let's see what topics we are going to review in this particular class. There are three large modules. The first is to understand what is the structural organization that occurs in proteins in terms of amino acids, the secondary structures that make up these amino acids, as well as what experiments are used to determine these three-dimensional configurations. As a second point, we will now start from that information that we explore and understand how it is that we as humans organize all the diversity of proteins in different classifications at the sequence level, at the structure level, and thus have a better organization of this structural diversity and sequence diversity. And finally, we will explore properly what characteristics this protein universe has, if there are some classes that are overrepresented, underrepresented, and how dynamic this particular landscape is. So let's start with the structural organization part present in all protein structures. Here we must obviously recognize that when one talks only about proteins, it seems easier to associate it with what food intake is, whether from meat, of legumes, or any other food with high protein content, or also when we talk about them. We think of these food supplements associated with a fitness lifestyle. But really, when we talk about proteins, we talk about macromolecules that are found inside all cells and that are capable of developing many functions, and that are part of the structure of all cells, and also provide them with their functions in themselves. Molecular entities that are actually quite dynamic, as you can see here in this video, where they are practically rotating and changing their three-dimensional configurations in space, although we usually associate them with static representations. But in reality, when we talk about proteins, we are talking about this type of macromolecules. And here what we can practically observe is the enormous structural diversity that exists in this type of macromolecules. Some are precisely responsible for providing structural support to the cell, or for establishing communications between different environments, between different molecules. Or others, such as perhaps it could be precisely this motor that we have just observed that provides movement capabilities to cells such as bacteria. Or we even have macromolecular complexes, as large as this could be associated with what is the entrance to what is the cell nucleus and access the DNA, which is the nuclear pore complex, which has to meet many specificities because obviously not everything has to pass to the nucleus, which is where the genetic information is. We even have enormous complexes, with millions of atoms, which could be centrioles or the centrosome, which are structures that are precisely assembled during the process of cell division, and that practically regulate this entire process of duplication and replication of genetic information. As such, really all proteins are found within this universe of biopolymers. And these molecules are characterized by 5 major properties, which could be organization by complementarity, which is reactivity in an aqueous medium, presence of chirality, which is a different behavior depending on its three-dimensional arrangement in space. As a fourth point, we have that they can be synthesized through dehydration reactions, as well as the presence of intermediate structural states mediated by bonds with phosphorus atoms. And as a fifth element, we have that these molecular entities also present a great diversity of three-dimensional conformations that actually represent different states of what we call folding, which is this three-dimensional configuration in space, which precisely seeks to protect the protein from chemical reactions such as hydrolysis, among others. And obviously these biopolymers do not exist alone. They exist together with other types of molecules that are very relevant to the ingredients of life, such as water, metals, or cofactors, that were precisely the ingredients to give rise to life, at least on this Earth. As the first element within this group of biopolymers, we obviously find what nucleotides are, right? The nitrogenous bases that make up the DNA chains that could be adenine, guanine, cytosine, thymine, and even here I failed to add what uracil is, which in general terms we talk about the functions that this type of biopolymers fulfill is practically the storage, management, as well as the propagation of genetic information, and that as ancillary functions they are even capable of performing a catalysis that is relatively simple and not so elaborate. As the second type of biopolymers that constitute life, we find precisely amino acids, which are the components of proteins. In particular, amino acids are characterized by being able to carry out catalysis that is now relatively much more complex than that carried out by nucleotides. They are also responsible for tasks of structural and functional regulation, and also provide structural support to be able to give this corporality to all the cells and their various forms that exist. Here it is very important to know some bonds or interactions that exist between the atoms that make up the residues. Any residue, as you can see in this histogram, is composed of several atoms or groups of atoms. In particular, what is the amino group is relevant, which is here a nitrogen united with 3 hydrogens, a carboxyl group, which is a carbon united with 2 oxygens and 1 hydrogen. On the other hand, we find simply a hydrogen, and on the other hand, we find what is known as a radical group, or side chains, which can be a multitude of atoms that precisely provide a specific behavior to each of the amino acid that distinguishes them as such. So we then find a diversity that an amino acid due to this particular arrangement that exists within this radical group could be called alanine, glycine, cysteine, depending on the type of atoms that exist in this particular radical group. And here an interesting property of amino acids is that they can precisely be concatenated and create increasingly complex structures. And this occurs through what is known as the peptide bond, a type of chemical bond that is covalent, which is one of the strongest chemical bonds that exist in chemistry in general, and that is precisely responsible for unifying 2 or more residues into multiple chains that already configure a more elaborate atomic structure. Here also another notation that is relevant to make is the nomenclature that we usually use to be able to refer to the 2 carbons that make up any type of amino acid. As you can see, there are 2, but what would be the central carbon, since it is where the amino group, the carboxyl, the hydrogen, and the radical are connected, is specifically known as the alpha carbon. And the other carbons that could be associated precisely with the radical groups, since they will be called by other Greek letter names, such as beta, gamma, etc. So these are the molecular interactions that really constitute the amino acids, and that precisely allow them to be concatenated to create increasingly complex structures. And as a third type of biopolymers, we find polysaccharides, which they are really responsible for, we could say, in terms of performing energy storage functions, as well as signal translation, and also providing certain structural support. These 3 ingredients were really the ones that diversified and were made from multiple interactions to really constitute what we know as life. For example, here what we find is a cell of a bacteria called mycoplasma, and in what is the lipid membrane, we find inserted in green multiple proteins that are responsible for both the structure and the functions that this cell can perform. And really, the issue of defining life is quite complex, but something that we can say that could define what life is that at least these 6 properties are present together. We are talking about the fact that to talk about life, we have to have a spatial and temporal order, which is evidently not provided by the cell. There is also talk of growth, replication, self-organization, and perhaps more importantly of Darwinian evolution. That is, it represents this type of matter in a historical way. And finally, sequences of chemical reactions can be carried out in this controlled space. So let's say that at least these 6 properties we can think are associated with life. But obviously, the discussion of what is life or what is not life falls very far from the discussions that we can address here in these sessions, and that obviously lead to more others to discuss. Something relevant here is knowing what is known as the central dogma of molecular biology, which is this communication that exists within the cell to transfer information from DNA to the creation of a protein. So as you usually know, we can find genes, and these are specific regions within the DNA. In particular, then, a process now occurs where the information is now transcribed into a chain of RNA, and in turn, this chain of RNA, through the translation process, eventually comes to constitute what is a protein, that is, this assembly of amino acids. Obviously, different processes also occur that allow replication, that is, where a feedback within this same type of molecules, or even that we can return the chain from RNA to DNA through what is known as reverse transcription. But in general, let's say that the central dogma that was raised in its first steps suggests that once the information reaches the protein's level, it can no longer influence again what would be the other levels that precede it. Something that perhaps is now a bit relatively deactored, because we have investigated other phenomena that effectively suggest that there is feedback even at the protein level. But let's say that this is the minimum necessary to understand how this step of information is taken from the DNA genes to a particular protein. And this happens thanks to what we know as the standard genetic code, which is the way evolution found to be able to organize amino acids by nucleotide sequences. So in particular, what is known as a codon is what is a particular 3-nucleotide configuration. And then, by translating this nucleotide codon, we will arrive at the formation of a particular amino acid. So in this case, when in a gene we find, for example, a cytosine together with another cytosine, and then perhaps another cytosine or a uracil or a guanine or an adenine, we can say that then this combination of 3 nucleotides will give us the residue that is a proline. This obviously occurs thanks to a whole machinery of other proteins that are precisely in charge of interpreting this information, and that evidently reach us with the results that are precisely the diversity of amino acids that we have. Which, well, let's say that in general terms they can be classified by their behavior, which is already polar, nonpolar, alkaline, or acidic with respect to this. And this can be understood in a better way with respect to knowing these side chains that are present in the amino acids are really those that determine the chemical and physical behavior that these amino acids carry out within the protein, which means that obviously, due to this particular configuration present in their side chains, they will have limited geometric or physico-chemical interactions or greater flexibility in this 3-dimensional space. So, for example, it can be relatively well illustrated here with different side chains of these amino acids, what the behavior is like in the form of like a dance floor. For example, those side chains that have 2 atoms present that have a positive and negative charge, such as this one in particular represented here with some red and blue spheres, because having negative charges that are opposite, it suggests that there is a high dynamism between them because they seek to maintain a certain stability. But when there are no charges, as could be the case with this amino acid, which is quite large and has a ring of carbons here, well, perhaps then its flexibility, its dynamism, is not so much in comparison to what would be precisely this side chain that does have these atoms that are of opposite charges, and that evidently provide it with this physical and chemical behavior. Here what we observe is precisely how these side chains of amino acids are characterized, which instead of writing them with their full name, we use two types of nomenclature. One that can be as little as 3 letters, and one that even compacts them even further to a single letter, in such a way that the alanine residue can also be described as the letters ALA, or simply with the letter A, or the amino acid, that is glycine, can be described simply with the letters GLI, or with the letter G. And thus practically all the 20 amino acids that are usually associated with what proteins are. And together they are known as proteinogenic amino acids. In this case, obviously based on these properties that we reviewed previously, such as this dynamism at a physical or chemical level, we can organize them into multiple categories. Perhaps the most relevant would be to organize them by their liking for aqueous environments. We could tell them that there are some who do not like water at all, and who therefore have a character that is more hydrophobic, and others who perhaps don't care or even have an affinity for water, which we can call hydrophilic. Obviously, this would be like a category like the most general one in which we can classify amino acids, but it is not the only one. We can also classify them according to the presence of charges, for example, positive or negative. If, for example, in the total configuration of the chain we can say if there is a certain polarity, or if there is no group with any charge that makes us think that they are greasier, or even if there is the presence of some aromatic rings within their chemical structures, as well as others that perhaps are more characteristic, and perhaps do not fit within all these classifications. So let it be clear that we can precisely organize amino acids according to their physical and chemical behavior into multiple categories. And this is relevant because precisely the three-dimensional interactions established by the side chains of the residues practically determine the conformation of the protein itself. That is, the three-dimensional structure that the protein itself adopts in space. And to do this, you have to know a concept that is key, which would be like the skeleton of the protein, or in English it translates as backbone, which really what it does is capture in general terms what the figure is, what is known as the folding of proteins. And this is because what is actually the peptide bond constitutes this part of the protein skeleton, and it is the side chains that provide this specific behavior to all specific proteins. So here, what we observe is that let's say we have this skeleton, and now we have to find all those side chains that satisfy this particular architecture of the skeleton. So if we adopt what is this classification of hydrophobic and hydrophilic residues, then we will see that at least in this configuration of amino acids, we can think that the atomic interaction is being stabilized, in the sense that here there are no clashes between amino acids, and that there is also physical-chemical compatibility in such a way that the charges inside are not repelling. And obviously, we could think that these side chains assure us a certain structural stability. But not so when in this case we observe that there are residues that have very short side chains, which is causing in the first instance a void within the protein, which is almost not possible because obviously the protein seeks to be relatively stable. But now we also find interactions that are not usually associated with what is inside the protein, given that in this case we have two amino acids that are hydrophilic, which should be outside the protein and not inside the protein. Inside, we essentially want there to be protein. Hydrophobic amino acids and evidently hydrophilic on the outside, since they are dissolved in the cell, which is a harassing environment. And as another example, It is now evidently very clear that this configuration of side chains is not possible here either because they are colliding precisely. These rings corresponding to each of its amino acids, and therefore it is not physically possible that they are actually found in this configuration within the protein. So now we see that it is clear that there is both physical and chemical compatibility of the side chains that configure precisely what the shape of any protein is. And what this really represents is, it is a set of molecular interactions that we can interpret as a kind of molecular glue that really configures the global structure of the protein. And obviously there are multiple molecular interactions, such as these examples. For example, hydrogen bonds that occur precisely through interactions of atoms that are electronegative, which are actually quite famous, since they are the ones that usually establish, well, assert stability in what is known as the secondary structures that we are going to review below. There are also others such as van der Waals interactions, which are relatively weak interactions. In fact, if you notice, the vast majority of the molecular interactions that I present here are of the non-covalent type, and only a few, such as disulfide bonds, which are these covalent bonds that occur within cysteine residues, are considerably strong. Or even the interactions that a protein may have with respect to a molecule that sticks to it, which we usually call a ligand, because, in the particular case of this, there are some ligands that do interact covalently with the protein and they no longer tend to separate from it. They remain completely stuck to it, and it is already quite difficult to remove them from within the protein. Even another interaction that is also quite relevant is what coordinate ions are, which are practically metals, which by giving up one of these ions, the electrons, in order to establish this type of coordinated covalent bond, a relatively strong atomic interaction is also established. But there are others that are obviously non-covalent interactions, such as covalent bonds, sialynes or ionic bonds that precisely occur through residues that have precisely charges that attract each other, including also interactions that exist within residues that have these aromatic rings that also precisely by having this ring-shaped configuration provides all these properties related to the renewal between multiple things, right? And here another interaction that is also relevant, but that is not really due to the presence of the waste explicitly, is the hydrophobic force generated by this entire environment that is aqueous. That is, the protein evidently seeks to have residues in its center that are hydrophobic and residues that are hydrophilic on its surface. So the presence of water is also a factor that also contributes to all the molecular interactions that stabilize the protein. Something that, for example, we already see in what would be this particular diagram, where again in gray what we see is the representation of the skeleton of the protein, the backbone in particular, and we find multiple molecular interactions that what they do is establish this particular configuration and make it stable. Here what we see, for example, are hydrophobic interactions. We also find salt bridges, dipole-dipole interactions, van der Waals forces, hydrogen bonds, among several others, which together make the protein adopt its particular three-dimensional configuration. Its shape, then, and here's something that I do like to highlight, is precisely the relevance that water has in being able to make these three-dimensional arrangements of proteins, given that water is not usually given any relevance, at least in structural terms of the protein. Here what we observe are different water molecules, represented by these little red balls, as well as the side chains of the proteins. And these grids that we see in these graphs, it is known as the electron density, which is evidently the most probable region in which we can associate the presence of the electrons of what is the water molecule, or what would be the electrons of the side chains. And what we observe is that the presence of this particular configuration of these four water molecules together with these side chains, what they do is establish a network of interactions based on hydrogen bonds. What they really do is contribute a geometric contribution to the protein, so that it is stabilized in a particular way. Here we even find another geometric coordination mediated by three water molecules that establish hydrogen bonds with two residues that are quite far away. But thanks to these molecules, they establish a certain communication in the geometric sense. So then it is clear that the presence of water is also decisive for the structure of proteins, something that is usually not addressed as much as perhaps I would be interested. Even if you think that we already know many of the molecular interactions that stabilize the structure of proteins, it seems that not and that perhaps there is still a new theory that we have to discover and investigate what this new molecular interaction that was only discovered in 2021 could be like, which the authors suggest that we can call a new interaction called the NOS bridge, which is an interaction mediated by lysine residues together with another cysteine residue in the presence of oxygen, in such a way that evidently a bridge is made between the nitrogen, the oxygen, and the sulfur atom, which gives it precisely the name NOSBRATCH. In this case, even when investigating how present this type of interactions is, well, they found them even in practically all domains of life, including us, and that, well, although it occurs under quite specific conditions, it is relevant in terms of the catalytic functions that can occur in proteins. And even when exploring it and how it is seen in its atomic representation in this particular protein of this bacteria, we see that these electron density maps are indeed found here, and here is this beautiful NOS bridge, where precisely here is the oxygen, here is the nitrogen, and here is the sulfur atom that constitute the bridge. Here, something relevant to understand what each of the amino acids is that they obviously have geometric limitations, which is given in part by the freedom they have to rotate in space each of the angles that constitute the amino acid itself, which is known as the dihedral angles of proteins. What does this mean? Oh, here what you see is an amino acid, which in this case is isoleucine. Again, here what we're looking at as the central carbon atom. This is the alpha carbon. This would be the other carbon within what is the general structure of the amino acid. In this case, we have this hydrogen that is not represented in the scheme in a conventional way, and here it would be the radical group that corresponds to isoleucine. On this side, we have the amino group, and in particular, what is the angle between the alpha carbon with respect to the amino group is known as the phi angle and the angle that occurs within what the alpha carbon is with respect to the carboxyl group. It is known as the psi angle, and these obviously have their level of rotation. Their degrees of freedom that they can have here is precisely what I was telling you. This would be the alpha carbon, and in this case it would be the beta carbon, the carbon, other letters of the Greek alphabet, and even what this angle is that occurs now in the side chains. It also has a specific nomenclature that is used, which in this case we simply call them chi. In the particular case of isoleucine, which has this specific side chain, it only has these two angles, which would be chi1 and chi2. But there are some residues that, due to their side chains, may only have one chi1, or some will have up to chi3, depending on what type of amino acid it is in particular. And we can see this precisely with this lannan bipeptide, where what is represented are precisely these essential angles that are pi and psi. And as you can see, they establish certain geometric limits in terms of the rotation of these angles that allow precisely this flexibility for the amino acid to move in space, right? There are some configurations that, let's put it this way, are prohibited because then there's a collision between atoms, which cannot happen because obviously they cannot occupy the same space. And so that determines, in part, the configuration that the protein as a whole can take. To be a little clearer in the representation of what the chi angles would be, here we have two other examples with what is amino acid K, which is a lysine, or with amino acid I, which is a tyrosine, where what we observe is that this amino acid, which is one of the longest side chains that we know, has up to 1, 2, 3, 4 chi angles of freedom, which are also represented in this diagram above. You can see them here, chi 1, chi 2, chi 3, and chi 4. In the case of the tyrosine residue, it simply only has 2, and the angles of a group such as a ring are not considered, since they do not have this flexibility that they would possess in this sense. Well, some carbon chains that are not in a ring configuration. And these geometric representations are usually graphed or represented with a particular type of graph that is called Ramachandran graphs, in honor of the researcher who investigated them, which can be graphed both for the side chains and for the amino acids themselves associated with what is the skeleton or backbone of the protein. Practically what they do is graph the numerical value of the angles that are possible to be adopted geometrically in such a way that they are consistent and do not collide. Then these regions of probabilistic density are formed where it is likely to find a particular configuration of pi and psi angles, and other regions where it will not be geometrically possible to find those combinations of angles in space. That is, there are precisely geometric limitations to establish three-dimensional configuration in amino acids. And these geometric limitations are precisely one of the structural bases that allow us to organize proteins at multiple levels, so that we as people can understand them in an easier way. What we are referring to when we talk about the primary structure of the protein, we are actually talking about its amino acid sequence, represented here as lysine, histidine, arginine, etc. When we talk about the secondary structure of proteins, we talk about these three-dimensional arrangements that are characteristic, which are usually called alpha helices or beta sheets, which really provide great stability to each of the residues and adopt this three-dimensional configuration within the protein. So within this type of secondary structures, we find what are known as turn regions. That is, regions that are not properly structured, as if they were the types of secondary structures that are precisely called alpha helix, which are stabilized by hydrogen bond interactions between adjacent residues within this particular helix. And we also have the secondary structures that are known precisely as beta sheets, which due to their configuration in space or their orientation and direction, well, we can say that in this case this beta with respect to this other beta are anti-parallel, as you can see by their direction, and in this case this other beta and this other beta are parallel, given that they do follow the same direction. These would be the secondary structures of a protein. The tertiary structure, we could properly say, is what the entire three-dimensional arrangement of the protein is, its shape itself, and that even these proteins can be assembled with other proteins, each with their own tertiary structures. And that now that particular set is known as the quaternary structure of proteins. This is just an outline that allows us as people better understand your level of organization in structural terms. And this was mainly investigated by multiple people, but perhaps the one who is really responsible for the discovery or characterization of the presence of these regions of unfolded alpha, helix, and beta secondary structures is due to this group of Linus Pauling, Robert Corey, and Herman Branson in 1951. That is, even a few years shortly before the characterization of the three-dimensional structure of DNA, who evidently explored all the atomic interactions present between these residues, such that they characterized it, and evidently saw that they adopt this configuration similar to a helix, or similar to precisely these beta sheets, or these foldings as if they were precisely a sheet in space, right, folded. And what they really suggest is that, well, obviously there are many geometric and physicochemical limits present in proteins, but one of the factors, or rather the most important factor that determines precisely these three-dimensional configurations, is precisely the peptide bond. As you can see here, there are two residues here, and precisely the peptide bond forms precisely a particular orientation in space, a plane that is known as the amide plane. where really what the pi angle and the psi are what play the leading role here in determining what the structure of the proteins would be. So then, usually when we evaluate the structures of proteins in these terms of angle, we always return to see these two as the main suspects to investigate, and later, we move on to the xi angles. Obviously, there are not only these three types of secondary structures. More has already been addressed and investigated in this regard, and currently approximately between 8 types to 9 types of secondary structures are recognized. Here, for example, there are different types of helices. For example, what is the alpha helix is also usually represented with a particular letter when we want to even further generalize our alphabet now in terms of secondary structures. But there is also another variety of helices, such as the 310 helix, the pi helix, or the kappa helix, and each one has a quite characteristic three-dimensional arrangement. And it is relevant to see, at least in terms of proportions, how these secondary structures are constituted in the protein database that we develop as humanity, which is the Protein Data Bank. This database consists of approximately 250,000 proteins, from which we obtain their atomic structures, that is, their three-dimensional positions in the space of each of the atoms that constitute them. What we observe is that, in general, these three categories associated with what the alpha helix is, what the turning regions are, or those which are the folded veins, are the ones that are the most abundant, and that these other variants of helices are really not as abundant in the protein that we have managed to investigate over several years of research, of scientific research. And thanks to algorithms such as AlphaFOD, we can now access the structural diversity present in multiple more proteins. For example, this set is relatively limited, which is 230,000 proteins, and by raising that number, now analyzing structural predictions made with AlphaFOD, to a number of approximately 1.5 million proteins present in databases such as UniProt, we find the same predominance of these three secondary structures, which is the alpha helix, the turns, and the folded vein. And also, even when we explore what are metagenomic sequences, that is, sequences associated with multiple environments such as rivers, estuaries, deserts, forests, soil, agricultural territories, among many others, we still find this proportion that the alpha helix, the gyres, and the folded veins are the most diverse in general terms. So these three are, let's say, the most representative when this type of structural level issues are usually addressed, particularly proteins. Something relevant here, returning precisely to the geometric limitations, is this movement, this flexibility that amino acids have. Here you see how they move in space through molecular dynamic simulations, which are like attempts to approximate, with physical-chemical bases, how these atoms move in space, precisely how their atoms move for proline, glycine, or arginine residues. As you can see, some vibrate more, some move faster than others depending on, for example, their charges, whether they are positive or negative or whether they have no charges in their structure. And this is very relevant because, coming back again to this type of graphs which, as I mentioned, are called Ramachandran graphs, when exploring the degrees of freedom that exist between the angles B and C, what we observe is that there are some probabilistic regions where we can find that the combination of these residues that practically constitute the backbone of the protein are associated precisely with this type of secondary structures in particular. So for example, these combinations at angle B, phi and psi, which are around these particular degrees, as they are usually associated with a secondary structure configuration of the folded beta type in particular. Or this other region here is usually associated with what would be a particular alpha helix from before. Then also the secondary structures, since they obviously have these bases of geometric limitations present. Due to the freedom of movement between these angles, which is the backbone here, as you can see, again, in this case the angle pi in particular is shown, and well, what we see is precisely the collision between what would be the side chains. something that would not be possible since the atoms cannot collide. Each one occupies a defined region in space, and therefore these geometric limitations are what? As they help establish this three-dimensional configuration of the protein as a whole. And it is really very relevant to understand in structural terms how a protein is configured, because if we understand its structure, we can also understand things like its function. For example, here we are going to address what is an enzyme called TEM1-beta-lactamase, which is an enzyme responsible for destroying a type of antibiotics called beta-lactam antibiotics, such as penicillin, cephalosporins, monobactams, or carbapenems. And in this particular case, we found that what would be the site where the destruction of this antibiotic occurs, which would be the catalytic site of the enzyme. There are multiple residues that do perform a task and that can attack using those electrons and destroy precisely the antibiotic. But here in yellow I put a very peculiar amino acid, which is a glycine. That in this case glycine does not have a side chain, a radical group that is very characteristic. But in this case, glycine simply has a hydrogen. And even, despite the fact that it only has one hydrogen, we could say that in this case applying a saying is very helpful to what does little in the way, since then, being so small. Well, it allows all the residues that are capable of carrying out some function because their atoms do have, I don't know, some with a positive or negative charge. Well, they can carry out the phenomenon of catalysis and destroy precisely the antibiotic, right? So, even though it only has one hydrogen, its role within the function of this enzyme called T1 has led it to be one of the relatively conserved amino acids in this protein due to its importance at the level, well, not to interfere with others. Even here we have another example with another antibiotic, whereby just changing a single atom, the effect that this antibiotic can have on other bacteria is completely changed. In this case, we're talking about an antibiotic called vancomycin, which recognizes a particular peptide present in the membranes of bacteria called DLA. And this interaction is mediated through 5 hydrogen bonds that we see here, indicated as dotted green lines. This is precisely the wing-wing peptide, and what is observed is that it reaches the antibiotic and precisely couples with these hydrogen bridges in order to affect the bacteria. But in the case that this peptide is modified because it corresponds to another species that does not have a LA, but in this case, it has a peptide called LACA. We see that then these 5 hydrogen bonds can no longer be established, and therefore the effectiveness of this antibiotic against bacteria that have this particular peptide exposed in their membrane decreases approximately 1,000 times, in relation to if it did have the interaction with the LA peptide. That is, just one atom that established a particular interaction, in this case, a hydrogen bond, resulted in the loss of 1,000 times the capacity of the antibiotic in question. Eliminate the bacteria, something that really matters to us here in another example. Again, we see in this case what the ion channels would be, in particular a potassium channel, such as the channels that we use within the neurons to be able to carry out this whole synapse phenomenon, and thus learn multiple concepts. And what we see is that so that this ion can pass to the other side of wherever the protein is, well, there has to be a reconfiguration of all the amino acids, such that a vacuum is precisely allowed here in the protein, such that our potassium ion then passes to the other side where it needs to be. Then they observe that there is a geometric reconfiguration in also physical-chemical terms of all the residues necessary for the potassium ion to pass through this protein, which evidently demonstrates the dynamism that exists in this type of atomic structures. Here is yet another example of what would be a protein called WAPE, which is a transporter of purines, that is, of a type of nucleotides, and what we observe is precisely these regions of electron density that would represent the structure of the protein. And now we are going to take a closer look at this structure where what we observe is an exquisite arrangement of its side chains, in such a way that they all occupy the space they must occupy to precisely establish its three-dimensional structure. So now we come to the point where we explore what are the experimental methods that are used to determine the atomic structure of proteins. Something that is quite detailed to address. It even deserves a course in itself, and that, however, we are not going to explore this in detail. We are simply going to explore relatively briefly what are the two main techniques that are currently used to determine the structure of a protein, which are the X-ray methods or the methods known as cryo-EM, and that, well, so that they can. To have more information about this, you can consult these reviews, which precisely offer more information on these topics in terms of experimental biology. So, generally speaking, as a first step, what we do is, first, design our project and think about what scale we are doing it on. There are scales that are relatively small, which are from 1 to 20 proteins, or when we want to massively evaluate 1,000 or even more than 1,000,000 proteins at a time. So the experimental design that we have to develop based on this, well, will change quite a bit. As a first step then, obviously what we have to get are the genes right. And these, well, we can already synthesize them in a way. Well, it would be very expensive to put the gene in some machine to assemble the particular gene for us in case we don't have it. But if we have it, we can simply do PCR experiments to amplify precisely this gene and be able to work more comfortably with it. Subsequently, now we have to move on to what the expression of this gene is in a particular species, something that at least in molecular biology is quite common to use. To the Escherichia coli bacteria, to be able to express our specific protein. And then here there is a point where we first have to investigate if our protein is capable of being expressed in E. coli, or if we have to use another bacteria or even a eukaryote, because perhaps everything that E. coli represents does not allow optimal expression of this particular protein, because perhaps it is more associated with systems that are of the type of eukaryotes and not prokaryotes. Subsequently, once we have managed to express our protein, we now have to be able to isolate and purify it to precisely only study this protein. And to do this, obviously, there are multiple experiments to be able to filter this and arrive precisely at protein extracts of high purity that only allow us to work with the protein that specifically interests us. And later, we do some preliminary tests that help us estimate what the stability or secondary structure compositions of this protein would be, and thus corroborate that we are working with the protein that we really want to work on before achieving its three-dimensional structure. And then, once these preliminary analyses of its three-dimensional configuration have been determined, we now move on to what is actually the determination of the atomic structure using these two techniques that I mentioned, which are X-ray crystallography as the main strategy that has been adopted by the structural biology community. But there are also other techniques such as cryo-EM, SAX, or NMR, each one for specific purposes and specific proteins in particular. And obviously once we can derive even more information from this three-dimensional structure, and obviously understand more about which residues would be capable of carrying out catalysis, of binding, of establishing interactions with other proteins, but that this information precisely derives from being able to obtain its three-dimensional structure previously. What we observe here is precisely what is the X-ray crystallography experiment like? Very briefly and simplified as much as possible. In this case, we start precisely from a solution that is pure of our protein, and now what we do is obtain a protein crystal, so that precisely the protein molecules are formed in a fixed pattern. And this is actually quite laborious given that not all proteins crystallize under the same conditions. Some require very specific conditions that we do not know a priori, and this procedure can actually take a couple of months, or, in the worst case, many years of research to be able to determine what conditions are optimal to crystallize a protein. We then talked about having to combine different concentrations of the solvents we use in order to obtain this crystal, which is not an easy thing. And well, as point number 2, now that we have our crystal that already includes our pure protein, now what we do is actually put it through X-rays. In this case, what is done is, let's put it this way, how to bombard it with these. What is called a diffraction pattern is obtained from how precisely these X-rays pass through the particular crystal and all the atomic information that exists in these crystals. So that with these diffraction patterns that are represented by these concentric circles, now we have to apply more algorithms such as these Fourier transformations among multiple other very complex algorithms that allow us to reconstruct the electron density maps, which is precisely like the probability that exists for this particular region, that the specific electron for an atom is positioned there. And so we build these precisely density maps. And from these density maps, we can determine each of the atomic positions on the X, Y, and Z axis of each of the atoms that contribute to the protein itself. And with this, we come to what is actually the construction of a protein model. This is how X-rays work. And now we are going to explore how the methods known as CRIOM work. In short, we start with a pure solution of our protein. And now what is done immediately is to freeze it completely, so that the dynamism that the proteins have within the cell. So it is completely frozen, and we can investigate its atomic positions in a much easier way. And again, now we pass our frozen sample of proteins in a beam of electrons, in such a way that by bombarding it with electrons, now we have, let's say it this way, like the shadows of what these proteins would be. And then from the shadows, we can collect a lot of information, given that these shadows, they are obtained from multiple angles of rotation and orientation of the protein, in such a way that it allows us to really infer more information within each of these perspectives. And it is from these images that are obtained of these particular shadows, let's call them that you can combine all this information and then reach a consensus of what the atomic location would be of precisely all the residues of the protein. This is how these two main techniques work, and obviously under certain particular circumstances, you can prefer the use of one technique over another in terms of money, time, as well as the complexity of the protein that you intend to crystallize in this sense. So here what we will observe is practically a summary of everything we have covered regarding the basic configuration of any residue, and then how these are concatenated to form the secondary and tertiary structures through molecular interactions such as hydrogen bonds, among multiple other things that actually constitute the protein in its entirety. There we see that a water molecule was released that precisely forms the peptide bond between two residues, and then here two more residues appear to obviously make more peptide bonds and concatenate more and more residues. Now, we have there an increasingly larger peptide, and now we have an entire sequence which I was precisely telling you was the characterization that is known as the primary structure of a protein that, when we find it in its three-dimensional arrangement in space, we can precisely talk of what is known as the secondary structure of proteins, which are usually stabilized by interactions such as hydrogen bonds, among others such as van der Waals forces, pi forces, among others. There we have an alpha helix in those black lines, precisely the hydrogen bonds. In this case, what we see is the concentration. Well, preparation of the amino acids in such a way that interactions of the type of hydrogen bonds are also established to form what would be the specific folded betas And now we have precisely the comparison of the two main secondary structures. Along with the other that now they also know are these spin regions, which are not properly associated with a characteristic three-dimensional configuration. And now, by combining precisely these regions of secondary structure represented by these helices, we arrive at what is the global shape of a specific protein, which is the tertiary structure. And in this case, by combining multiple proteins, each with its specific three-dimensional arrangement, which in this case are many duplicates of the same protein. We talk about what is the quaternary structure of a protein, that is, a protein complex. That's good, actually, because what happens is that these are representations that really help us understand how the interactions between protein atoms occur. Although these stabilizing hydrogen bond interactions do exist, proteins are actually interactions between atoms, and here what we see is a representation precisely of how they are. They break the bonds in a quite artistic way, and how new bonds are formed in these entities that remember they are not static, but are highly dynamic in the space that represents the cell, which is also a chaos, as we will investigate in the protein function module of this protein engineering course. And this way, we have of representing proteins in the form of arrows and spirals. It was really a very well calculated, very well thought out, very well developed interpretation by a researcher called Jane Richardson, who actually wrote an entire treatise, which is called The Anatomy and Taxonomy of the Structure of Proteins, which was based on even artistic inspirations, but also on the physical chemistry of the interactions that occur between residues, because she came to the conclusion that perhaps these representations of spirals and arrows, well, they are the most optimal to be able to communicate the three-dimensional configuration that the residues adopt in particular, and make it easier for us as people to interpret precisely this specific geometry of the proteins, which, well, to have more details of why he came to this conclusion, you can access this document which is freely accessible, and here we have precisely how proteins were modeled before. For example, John Kendrew, who was the first person in history to be able to crystallize a protein, there really was no consensus on how to communicate all the information about the atomic structure of proteins. So Kendrew at that time, which was 1957, took on the task of making a mock-up of his particular protein. Something that really, although we as biologists, specifically those who study proteins, perhaps found in a very captivating way how he managed to represent the atomic structure of his protein using these models based on plasticine and wooden sticks. Let's say that frankly it does seem like a set of feces, that even what is in the news media called The Guardian really called it the feces of the century as such. What this really represents is the structure of a protein, the first protein. Obviously that was in 1957, and now we already have multiple programs and multiple representations that we can use depending on the idea that we want to communicate to our public about what the structure of the protein is. So in this case, well, in less than a minute I can download a protein, load it into a program and obviously represent it, either in its configuration of pure atoms, in its configuration of secondary structures, or also of secondary structures, but now represented in a tubular form to share in an easier way how the geometry of the protein is arranged such that it constitutes these units of secondary structure. This is quite careful to choose, since obviously the type of representation will depend on the audience and the idea that we want to communicate, of proteins that we have to use. And for that we can use multiple programs such as Chimera and PyMol, which are the two most famous programs that exist to model and manipulate the structure of proteins. But there are also other programs such as MolStar or MolIstasco. I don't know what they want to call it. There are even other more modern programs that are based on three-dimensional rendering engines that are used to be able to make Pixar films, such as the program known as Blender, and that make quite beautiful representations of these animations that we have seen throughout this class, and in this case, to be able to model proteins using Blender, you can use this library called Molecular Notes, and something quite relevant or beautiful is that even all these programs have an API, a section that is pure Python code, which you also, instead of manipulating it with a graphical interface with the mouse and selecting each of the options manually, because if you know how to program and have many proteins to analyze, you can simply write a script based on the Python programming language and thus process its structures in an automated way in this sense, which is quite simple. Beneficial for us, however, here we must also remember that, although we explore these representations in the form of spirals and arrows, what we are really trying to model or represent with this type of figures is a protein that is super dynamic in real terms, and that, having been represented like this, it really helps us understand more about what the three-dimensional configuration of the protein is like. Well, perhaps it is also limiting us to the way in which we conceive proteins in such a way that there are even people who evidently think that a protein is completely stable, as is the crystal that we are manipulating in a program. Something that is not true. The protein, as we saw at the beginning, is super dynamic. It is in constant movement in practically any period of time, and so here is what they suggest to us in this perspective, is that we should really think of proteins as collections of atoms that are constantly establishing very dynamic interactions with each other, as well as with their environment, in order to perform their function. Although we see them as static structures here, there really is a lot of movement, something that is quite relevant to consider when we are thinking and reflecting about proteins in our work. So once all these concepts have been explored, we move on to the second part of the class, where we are going to explore how these structural classifications of proteins are actually constructed. And to do this, you obviously have to know the basic elements on which they are built. And to do this, let's first explore this protein, which, as you can see, is there in its three-dimensional arrangement, which we can represent in a two-dimensional way with a map that indicates the connectivity between its secondary structure elements. You can see here that the protein starts with an alpha helix, followed by a beta 1, 2, 3, 4, 5, with a color gradient from red to blue. So if we rotate our protein, we actually see that we start from our alpha helix, we continue through the red beta, then the orange, it now connects with the yellow, then with the green, and we end with the blue as indicated in this diagram. So now the question arises, does this new protein, whose connectivity of the secondary structure elements that it integrates is the same protein with respect to this other one up here? And this is where the concept called folding, or in English FOD, arises, which corresponds to the number, type, connectivity, and three-dimensional arrangement adopted by the secondary structure elements of proteins, in such a way that, since this connectivity between elements has been modified, usually by default, it is thought that they are two different proteins. And to prove the opposite, obviously much more detailed structural analyses must be carried out. But when we refer to this set of conditions, we can say that this is protein folding. The second essential concept to understand is precisely what is known as domain. And for this, we are going to address the hemoglobin protein, responsible for transporting oxygen in our blood. As you can see, the hemoglobin protein is actually made up of 4 other proteins, which are these 2 proteins that we see in orange and these other 2 in blue. Because these are proteins that are different, in this case we are talking about it being a protein complex, or we can also call it a multimer. And an even greater distinction can be made based on the identity of the copies that make up this multimer. So that if in this case different proteins constitute the hemoglobin protein, we can say that this multimer is now called heteromultimer, because the identity of the proteins that constitute it are different. If they were identical copies, and it was obviously a protein complex, we could also call it homomultimer, And when we are going to analyze what these orange proteins and this blue protein correspond to, they correspond to proteins that present the same type of folding, or at least that is how they were classified, which are known as the globin type folding, and each of them is called alpha globin and beta globin, which when comparing the amino acid sequence of each one, we found that their similarity is 42%, which clearly illustrates that these are different proteins and not copies, and therefore is consistent with what I told you that this is a heteromultimer. What would a homomultimer look like then? Well, then here it is quite clear or evident. Here what we see is a gene, and let's say in this gene each of these little red blocks encodes this particular type of folding. And then, this folding actually, when the complete gene is translated, encodes this protein as a whole, which the gene is found here with 11 repetitions of this specific protein, which corresponds to a bacteria called Burkhardia sapronensis. And you can see that the three-dimensional arrangement of the protein is already like circular or discoid in appearance, but is actually composed of duplications of the same subunit. Therefore, in this case, we can speak of it being effectively a homomultimer. As a third example, we are going to address one of the most abundant proteins on Earth, which is the Ruisco protein, without which things like photosynthesis would not exist. In this case, when we inspect the structure of the Ruisco protein, we can see that it has a kind of 4 spikes that are indicated in orange, and something that seems to be similar to a base on which these spikes are mounted. So now we are going to inspect in more detail the parts that make up each of these elements. It turns out that if we grab any of these 4 little orange beaks, it actually corresponds to the largest subunit of the rubisco, and if we grab one of the arrangements that are in blue, those are known as the minor rubisco subunits. And if we go into more detail about what type of folding these subunits constitute, we find that in what is the largest subunit of rubisco, there are actually 2 types of folding. One is known as the timbrel fold. They are connected here by some twist regions, and then it is connected to another domain that is called alphabet braids. When we inspect how many types of folding there are in what is the minor subunit of rubisco, in general we observe that there is only one, which is this one in color 100, which is known as ribulose 1,5-bisphosphate carboxylase oxygenase. So you can see that there is a kind of combination of multiple foldings that constitute the protein, and at the same time, they can evidently congregate in quaternary structures that make it still, because it becomes more complex. So in general terms, we can say that a domain corresponds to a single type of folding, although this has some exceptions that we will investigate at the end of this class. And now one of the most natural questions that can arise from this is how many domains can a protein have? And the answer is who knows. Actually, one of the largest proteins that has been known so far is this one called titin, which is actually responsible for part of the musculature. We have to be able to move all of our skeletal muscle, and which is composed of approximately 240 domains of the pseudokinase, FN3 or Ig-like type, which in this case form a protein of up to 35,000 amino acids in total. And if you're wondering, is this the largest protein we know of? The answer is no. We even know even larger proteins, which in this case is this one called pecacil, which is made up of 140 domains of this type, ACP, Ks, Kr, DH, ER, which in total represent approximately 45,000 residues. You can see that there are specific protein complexes in this case. Extremely large heteromultimers that are already on the order of thousands of amino acids, or even up to tens of thousands of amino acids. So it is evident that we have too much complexity. And then, how do we be able to be clear about what we are talking about and share our ideas in a clearer way? and then at least since 1958, one of the first publications within what would be the protein organization carried out in this case by Francis Crick. This fact is addressed right, where what it suggests is that we should have a kind of taxonomy of proteins that, by comparing them, we could better understand the species from which these proteins come, right, in such a way that this type of units really contain evolutionary information that could help us precisely to understand better what the evolution process is like. And this is because classification systems have actually emerged in multiple areas of science and human understanding. Chemistry obviously also has its periodic table that organizes the elements in terms of families and multiple other components. Mathematics still uses different types of numbers to define what space we are working in. We as biologists still classify all the biodiversity that exists in different categories based precisely on their evolutionary distance when comparing many of their genes, many of their proteins. And even physics also catalogs each of the particles that make up reality in a standard model. So it was really quite natural or consistent to think that even proteins also have a taxonomy. However, it was not really known how to construct this taxonomy. If we consider that myoglobin was the first protein to be crystallized using these, he was the second person to obtain a structure of a protein. So, we lacked enough data to be able to carry out this type of taxonomies, something that even John Kendrews himself recognized when he said that there seemed to be no regularities in this type of molecules, which made their cataloging, their ability to make a classification, much more complicated, which is also very consistent as molecular biology evolved, given that even since 1951, it was the first gene that could be sequenced by Friedrich Sanger, who explored for the first time what the sequence of a protein called insulin is like. That in this case, although perhaps now it seems moving to us or even somewhat funny, these types of electrophoresis tests were carried out where you had to date, in which spot on this plate corresponded to the weight of which amino acid, and thus you were identifying the identity of the complete protein sequence. But we had to analyze huge plates like this one here to really be able to reveal the identity of a sequence, something that now we simply send to sequence, and we have it practically in a file that weighs a few kilobytes, and that is already, frankly, automated. But at that time it required a lot of experimentation to arrive at just one sequence. And it was not until approximately 1964, for this publication, that it was a watershed in what was the recognition of proteins as molecular entities carrying evolutionary information, which was a proposal made by Emil Zuckerland and Linus Pauling, which, as I said, well, it was a watershed at least in the field of proteins, who said that the most informative phylogeny that we can build is, it can be carried out from molecules, and that because evolution occurs through the process of mutations, and that these mutations can have profound changes at much higher levels. That is, for example, if a mutation occurs in an important gene, well, perhaps you can die, right? So these macromolecules, that is, proteins, contain very good information regarding the history of organisms, regarding perhaps other sources of information that had been used until then, such as the anatomical part. Currently, obviously, these two types of information, such as anatomical or genetic information, are compatible. It is not that one is better than the other, but that they complement each other, and that is something that obviously every biologist must recognize. And you can obviously explore this in a much better way in these seminars held by Antonio Lascano at the National College, where he tells us more about how this procedure for recognizing the amount of evolutionary information that a protein itself can contain was given. something that they also discuss in this chapter of the book. So that even here they show several examples of how we can extract this type of evolutionary information by comparing proteins. And comparison is the key word here, in that really, at least within biology, almost nothing makes sense if it is not in the light of comparison. Here what we observe is that there are several proteins, several genes. In this case they are nucleotides, as you can see here A, C, G, T. But we can actually do the same type of analysis at the protein level. And then we observe that they are a gene that corresponds to different types of animals. We as humans are here, but we are accompanied by other types of primates. Then we are more closely related to other types of rodents that are also animals, and so on with other types of animals. And what we observe is that there is great diversity at the level of the nucleotide gene sequence, but that there is a strip here. I can observe that there is almost no variability regarding the arrangement of the nucleotides, which suggests that it is relatively well conserved despite the hundreds of years or thousands of years of evolution that exists within this group of animals. Therefore, we can think that this region plays some important role, but a priori we do not know if it could be related to what the structure of the protein would be, to the function of the protein, or to multiple aspects. But the idea here is that we can identify these kinds of interesting patterns that signal some evolutionary signal by comparing various proteins from multiple species. This was even taken up by the creator of the first protein database, whose name is Margaret Dayhope, which can really be considered together with her team, which was actually mainly made up of a group of women. That they really introduced what was the use of computers to be able to analyze practically all this diversity of proteins, and not do it in an artisanal way one by one, which would really limit us quite a bit in our analysis capabilities. And then we could create this type of evolutionary analysis, where we can see what type of species are closest to each other based on the analysis it provides us. In this case, a protein that is known as cytochrome C, which is a protein belonging to the electron transport chain that we have practically in many forms of life, which is to say, it is one of the key components in the generation of energy by any form of life, and therefore, when comparing these genes, we can see certain relationships between all types of animals and even with plants. And even the work of Margaret Dayford, which is really reflected in our first database with just 65 proteins at that time, allowed the identification of what are known as protein families and protein superfamilies, based on how much their sequences resemble each other at the residue level. So that a family of proteins, we can say that it shares a sequence identity with other proteins of the same family, of approximately more than 50%, and that when we talk about a superfamily in general, the proteins belonging to one of them would share between 30 to 40% of identity. And this obviously translates into diversity at the sequence level, but is also reflected at the level of the structure of the proteins, as we are going to see later in this class. And until then, consider that this was in 1965. It was not until 1976, almost 20 years later, that what would be the first structural classification of proteins appeared. That until then, returning to what were the first works carried out by John Kendrew and Max Perutz, we would only have managed to compile around 31 protein structures. And so in this work, done by Michael Leavitt and Cyrus Clocha, when analyzing these approximately 30 protein structures, they managed to classify, let's say, this structural universe into 4 large categories. One that are, therefore, structures or proteins that are composed of pure alpha helical regions. Others that are composed of pure fold betas. Others that are with alpha-beta or beta-alpha patterns in an interspersed way. That is, here you can see how an alpha is interspersed, and here a beta, or even a beta and an alpha. And then regions that are completely separated from alphas and also from folded betas, but both types of secondary structures are present in the same protein. And so here you have a kind of catalog of what these types of proteins look like. Here it is quite clear to observe that proteins mainly with an alpha helical position have this quite characteristic Rigellite shape. The proteins that have mainly pure beta chains, well, you can also see that it is quite distinguishable. Even what is the composition of alphas and betas? Well, we also have some patterns here, as in this case where alpha beta, alpha beta, alpha beta, alpha beta are connected, or also regions where there are pure betas and here also pure alphas. So now the question is, well, what is the information that we are going to use as a basis to be able to generate our protein classifications? We can take the proteins at the sequence level and generate a multiple alignment of sequences, as we can see here, in a way that allows us to observe that there are some residues that are conserved throughout evolution, and that perhaps there are some regions where there is more variability, where we find more mutations that are also interesting to investigate. However, the question then becomes, is this information enough to construct a classification? In this case, if we now consider the structures, well, we are missing out on all the three-dimensional information they offer us. Just as if we only selected what the structure is, we are also losing the sequence information that we have when comparing a greater number of protein sequences at that time, compared to the only 30 structures that were available at that time. Or even, what do we consider relevant within the protein structure? Elements that are quite conserved among all structures, their variability. That is, what elements are relevant to be able to build our classifications. And then, let's say that in order not to complicate our lives, we practically made two great schemes. Classifications that are built on a strong structural basis, and classifications that are built on a strong sequence basis. And here are several examples of what they would be, although currently there is already a resolution at the level of sequence and structures, so that we would not think that they are completely unrelated. But it is important to think that its basis is more structural, and the other has a basis more at the sequence level. In the case of the classification systems that we currently consider at the structure level, we have classification systems such as CAD, SCOPE, or ECOT. Although obviously there are also other systems, these are the most used. And at the sequence level, we also have classification systems such as PFAM, CDD, or superfamily. And obviously each one says, I'm going to use these types of algorithms. I'm going to use these evaluation criteria to build my classification of proteins and say, these are all alpha, these are all beta, these are alpha, beta, etc. Something that we are going to explore in more detail, so that it is understood and clear enough how these structural classifications work. So here what we have is the comparison of the structural structures of some called fuchsines. And in particular, what we have is this protein, but present in multiple organisms. What we observe here is that the structure is present in archaea, in algae, in viruses, even in animals such as the nematode C. elegans. And this is quite relevant because among this type of life forms, there really are thousands of years of evolution, and as we observe, the protein, even its shape, its structure, well, it is quite similar despite billions of years of evolution. And then the question now that also arises naturally is, well, how much does the structure change throughout evolution compared to how much does the sequence change throughout evolution? And then in this particular research, the title is quite declarative of the conclusions, which is that the structure of a protein becomes between 3 to 10 times more conserved than a sequence. As you can see, despite the millions of years of evolution that occurred, the shape is really preserved, which is even quite artistically represented by this particular quote, which is from Holm and Chryssander, who point out that comparing the shapes of proteins could be seen as using a large telescope that allows us to explore the universe, because it opens the door to being able to observe, detect the deepest evolutionary relationships that exist despite the billions of years of evolution that could have occurred between different forms of life. What does this mean? Well, when we explore what the conservation of structure is with respect to the conservation of sequence, we find quite interesting patterns. Let's say we have several sequences, and when we reach a limit of 50% shared sequence identity between two proteins. In general, What is observed is that around 90% of what would be the conserved regions at the structural level of the protein are shared between these two proteins. And even when we lower the sequence identity between two proteins as low as 20% sequence identity, we can find that even their structure is conserved approximately between 40% and in other cases up to 98%. Despite the fact that there is evidently a considerable difference at the level of their amino acid sequence. And if you notice, these observations made, which now have both information at the sequence and structure levels, curiously seem to be relatively consistent with what Margaret Dayhoff's group found in order to delimit what protein families, or protein superfamilies, were. So it means that even with so little information that team had, well, he was able to make quite good observations to reflect this consistency between what is diversity at the sequence level and diversity at the structure level. And why is structure in some proteins more conserved than others? This is precisely due to the rate of evolution that these proteins have over time. Something that even helped us build an idea that allows us precisely to reconstruct the evolutionary history of life, which is known as the molecular clock. Because there are some proteins that evolve relatively faster, or rather, evolve is incorrectly stated, but they change rather. There are some proteins that change more slowly, and there are other proteins that change much faster due to the number of mutations that can be found. So here we have, for example, some proteins found in some animals, particularly vertebrates, insects. You can see up to here some mammals like us. And we find here on the y-axis the rate of changes that exist per 100 amino acids present in proteins. And here then, the thousands of years of divergence that exist, billions of years that exist of divergence in evolutionary time. So that, for example, when we observe how the cytochrome C protein behaves, its mutation rate is considerably lower compared to hemoglobin, which can also be compared with other species, or other peptides like these called fibrinopeptides, which due to their context, where they perform their function, is obviously perhaps relatively less relevant. The function that a protein as important as cytochrome C would be playing, which as I mentioned belongs to this electron transport chain that is practically one of the energy-generating centers of any form of life. So we could think that also due to the functional relevance of the type of proteins, perhaps they could be more or less conserved. Although in order to really conclude that in a conclusive way, in a categorical way, obviously quite detailed, evolutionary analyses must be carried out. Here the idea is that thanks to this molecular clock, we can also catalog all biodiversity in terms of life forms in different categories, which we have called domain, phylum, class, order, family, genus, and species, depending on how similar the genes that constitute each of the taxonomic units belonging to these large categories are. The categories are precisely generated. The idea is that there are different markers that will also offer more or less information to be able to resolve evolutionary relationships that are both deeper or more recent. So that, for example, we also use specific genes that mutate much faster, and that obviously have more information to be able to discriminate between a diversification event at the level of species whose divergence event occurred recently, millions of years ago. But when we want to go to evolutionary relationships of billions of years, which could perhaps be reflected at the level of what would be the largest categories that we build in the taxonomic hierarchy, such as phylum, class, or order, well, now we need another type of genetic information in a way that allows us to access that evolutionary depth and thus discern the relationships. Because if we take genetic elements that offer us a greater relationship at the species level, here perhaps we will find greater variability that will no longer be reflected as noise, and that will not allow us to distinguish reliable relationships in evolutionary terms. So let it be clear that there are different evolutionary markers that are complementary and that allow us to build these taxonomic hierarchies that we as people create in order to better understand and catalog the different forms of life. Just that these topics really correspond more to what is known as phylogenetics, something that we are not going to address in detail in this course because we are going to focus on proteins. But if you are looking to learn a little more about what phylogenetics and evolution is, you can access this course developed by a colleague who is Dr. Maria Segovia, which is also a completely free course and is precisely based on what phylogenetics and phylogenetic inference in general is in order to build this type of evolutionary analysis. Likewise, another colleague from the National Autonomous University of Mexico invited me to give a class where I also addressed these types of topics, also using metagenomic information derived from the analysis of microbes, which is also available as a video on YouTube, so that they can address either of these two resources in case they want to learn the topic of phylogenetics in greater detail. So now we address what is, let's say, the necessary requirements to be able to compare structures or also sequences. They are both valid in these cases. As a first step, what we need is something quite clear, is a representation of the object we are studying. In the case of sequences, it is quite clear that we are talking about an alphabet of 20 amino acids in total, and in the case of proteins, it is more common to use toxic atomic positions of each of the residuals in the form of X, Y, and Z coordinates. The second element we need is a function that allows us to evaluate how similar our objects that we are comparing are. As a third element, we now need a comparison algorithm that actually serves as an optimization method so that, when executed, we get this similarity between the bodies we are studying. And as a fourth element, we obviously need a set of premises, of rules that categorically mark, although perhaps the behavior is more continuous, that beyond this threshold we are talking about two different entities, or that approximates that this could be more probable. So here really one of the things that must be recognized regarding comparison methods, whether at the structure level or at the sequence level, is that there really is no consensus on what we are talking about when we talk about structural similarity. However, although this consensus does not exist, when we observe it, it also seems quite intuitive. It is a question that we share in many areas of science, that even, for example, when you ask a physicist what time is, he will not be able to tell you in a completely clear way, but we all recognize the passage of time from our perception of reality. So here one of the things we can address is what kind of representation we can adapt in terms of the structure of the protein. We can adapt a completely rigid representation, or recognize that these molecules are, well, really super dynamic in real terms, but that, well, now this makes the analysis that we can develop with, well, the algorithms and the computing capacity that we have much more difficult, right? Since, well, we do not have this type of information for all the proteins that we know and analyze them as well, they represent greater computational cost, as well as, well, new paradigms that we have to develop to compare entities that are now dynamic, right? As a second point, now we can also even represent the protein not as three-dimensional structures, but as two-dimensional information, which in this case is part of a three-dimensional structure, no more than now let's say that we consider the neighbors that are if we look at a specific residue and verify if there is some type of contact between these residues, in such a way that then, if there is a contact between these parents of residues, we place a black cell here that is indicating to us that there's a contact, right? So let's say here we have, I don't know, a protein with 100 residues in its sequence. So then here it is, I don't know, 10, 20, 30, 40, 50 up to 100. This is what it would be telling us, is that approximately residue number 30 is in contact with residue number 10. And now we have a two-dimensional representation of the three-dimensional structure of proteins. Or we can even extract information, not only like this two-dimensional representation, but even build graphs of proteins that also tell us if they are in contact or not, because the residues of a specific structure, this is not the only representation of a protein structure. There are actually multiple possible representations of this structure which can be like point clouds of atoms, one dot for each residue, consider only what the surface of the protein is. Consider it now in volumetric terms. Consider it now as a geometric perspective that is known as Voronoi tessellations. Precisely construct a protein graph as we already explored, or only consider its secondary structure elements. Here, as you can see, there are multiple representations for the same object, which is also important to consider when you are developing your structure comparison algorithm, because what you are looking for is, well, obtain a representation that is most useful to be able to distinguish between types of proteins, and we can even do a general analysis in such a way that we take all the proteins, analyze them with various algorithms and extract statistics in such a way that there are even options. Well, there are research that do this type of analysis, and that, instead of representing them with these more three-dimensional perspectives, we now represent them as n-dimensional. Numerical vectors of I don't know, 128. 612, 1024 dimensions, which when analyzed using algorithms, well, now we have a vector representation as it works with the neural networks that we previously explored. Then they find that there is enormous diversity in the way we can represent the same object, and that there are even those who suggest that we should not take what the folds are as the unit of recognition, but that we can even go to higher levels, even smaller, at the level of what are known as structural signatures or motifs, which are small configurations of structure that happen to be shared between multiple proteins that are not evolutionarily related, but these small fragments are present and that precisely serve as signatures at the structure level. No more than even the way we infer these fragments, these structural signatures. It also varies from algorithm to algorithm. What you can see here is that there are three types of suggestion. In this case, the one is called fragments, this one is called themes, this one is called rifle. And in each one, they find a different number of structural reasons. What I intend to communicate here is that really every method always uses different representations, different qualification methods with their corresponding limits, as well as different optimization methods, which make them not strictly comparable, and which perhaps complicate a little what this whole procedure of being able to catalog the diversity of proteins at the structure level or at the sequence level is. And although it is necessary to recognize the complexity behind being able to compare atomic structures that in themselves are dynamic entities, it is evident that we also need tools to be able to operate and compare them, right? So here on this slide, we are going to present what are the main metrics that are usually found in the scientific literature to be able to compare protein structures, where perhaps the most common, the most famous is precisely this one, called RMCD, which means root mean square deviation, and which in summary is that once you have overlapped the atomic structures of two proteins, you measure what would be the Euclidean distance between their corresponding atoms, values, close to zero being indicative, which means structures that are more similar to each other. And when it obviously takes a value that can be positive or infinite, it means that they are structures that are more dissimilar to each other. In this sense, when we have a range between 2 to 3 armstrongs of distance between these two protein structures, we can say that these are structures that are quite similar to each other. But as this difference increases to 10 armstrongs, perhaps 20 armstrongs, well, obviously we're talking about there being no compatibility at a structure level between these two proteins that we are comparing. However, this metric has more particular conditions in which to be applied, since it really finds its greatest usefulness when we compare structures that we previously know already have an evolutionary relationship. That is, they are homologous structures that come from a common ancestor. And something that's really interesting is that when we begin to investigate how the rest of the metrics work to compare protein structures, we found that practically many of them use the RMCD as a base metric, but that they act as certain complements or add complexity to the matter to sophisticate their metrics, and thus be able to be more sensitive in a certain way. Here in this case in the diagram, what you see is a red protein compared to a blue one, and that by overlapping their structures, we can measure the distance between the alpha carbons to evaluate if they really are structures that are similar to each other. Another second metric that, as I already mentioned, is also part of what the RMCD is this one called TM score, which is precisely more sensitive given that it considers when comparing. Also, what is the length of the protein? Which is obviously really necessary because comparing a protein of 100 amino acids against one of 110 amino acids is not the same as comparing one of 150 amino acids against one of 500 amino acids. So, really considering what protein length is also provides relevant information. And in this sense, the authors suggest that how can we determine that two folds are similar based on this metric? And to do this, they define this metric in a range from 0 to 1, using a value of 0.5 as a fairly decisive cutoff point, such that if two structures that we are comparing share a value equal to or greater than this value of 0.5, we can say that it is a fairly similar folding. And obviously, the higher this value is, the more similar the folds will be. Something that we observe on the This similarity is due entirely to chance, and not really to a structural similarity, either due to the effects of these ancestor-descendant dynamics. So this is actually one of the most used metrics since it is also quite optimized, runs quite fast, and what it really helps us with is to be able to evaluate whether two structures share the same folding pattern in global terms. Let's say that with respect to the RMCD metric, this metric is more general, and the other is more specific because it is usually recommended or more sensitive in cases where we are precisely talking about structures that we already know are homologous. In this case, with the TM score, we can compare proteins that may have nothing to do with it, and it will be a very useful tool that will help us determine if our query protein it is structurally similar to a database of protein structures. As a third metric, it is one that was even used in the development of the AlphaFold algorithm, which is called GDT, which in English is called Global Distance Test. And in summary, this also involves the activity of being able to overlap the structure of two proteins. Only in this case it looks at different models. Distance intervals measured in Armstrongs, which in these categories of 1 Armstrong distance, 2, 4, up to 8 Armstrong distance, evaluates which fractions are within these four categories in common between both proteins in such a way that it is defined with values between 0 to 100, being values indicative of a similar folding between greater than 50 to 70%, and that as this value evidently becomes larger, the proteins will be noticeably more similar to each other. This metric is generally used to evaluate, like the TM score, what would be in general terms the structural similarity between two proteins. And as the last metric, which is also present in the AlphaFold system, is the LDDT, which in summary what it does is something relatively similar to what the GDT would be, however this, and above all that it is its most relevant attribute, does not need to overlap two structures in itself. It operates independently, and in this case what it does is look at the residues in its vicinity, up to a radius of 15 away, and then evaluates what fraction of these amino acids preserve their distances, and thus gives us a score to precisely evaluate the preservation of these distances between both proteins. In this case, it finds defined values from 0 to 1, being values indicative of a well-structured protein that would therefore have a similarity at a structural level with respect to the one we are comparing, approximately 0.7. So these proteins are the ones that are usually used, especially the RMSD, and secondly, the TMSCORE, when we are making more general comparisons. However, if you are looking to learn a little more about what this procedure of being able to compare atomic structures of proteins is like, you can check out this fairly detailed review, where you can see that there really is a diversity of structural comparison metrics, whether three-dimensional, two-dimensional, or even one-dimensional, that really recognize the diversity of what proteins represent, so that there are methods that include either patterns that are quite rigid for proteins, or consider them more flexible and elastic. And here the important thing is that really all the metrics we have to compare protein structures are based on differences and representations, different methods of scoring these similarities, as well as algorithms that optimize this process of determining structural similarity. Now we move on to what similarity search is, but now at the sequence level, where in general terms and in a fairly summary way, we have two general types of being able to compare sequences in this sense. We have one called a global search, which does, as its name suggests, compare the sequence in its entirety with respect to another set of sequences. And then the other aspect, which is the local comparison algorithms. What they do is evidently not consider the entire sequence, but rather small fragments that help the search become more optimal and more computationally optimized. In this sense, in each of these cases we have special algorithms, such as the Needlerman-Wilch algorithm, specifically for global alignments, or the Smith-Waterman algorithm for local alignments. In this case, global alignments are usually applied when one wants to do phylogenetic analyses. And therefore, we already know that we are analyzing sequences that have a common ancestor, that is, that they are homologous. And as examples of algorithms that can be used within this global alignment, there are some such as MAFT, MOSCUL, or TCOFI, which I remind you, are usually associated with what are phylogenetic analyses. However, when we want to buy a protein, we don't really know who it looks like, and this means that we have to search one protein against billions of proteins. Then we need more generalist strategies that do not start from the premise that our sequences are homologous. In this case, ideally, local search algorithms are applied, which in this case precisely do not start from this premise that they are homologous sequences that we are comparing, because what they really intend to do is a fairly global search to see what protein my protein that I want to investigate resembles. In this sense, the most famous algorithm historically has been BLAST, although there are actually much more optimized versions currently that run much faster than BLAST, which are called Diamond or MMSX. And here in general, the strategy that we can say that follows in both algorithms is relatively a divide and conquer strategy, given that really a fairly brief and simplified summary of how they operate. It's really doing like subfragments and comparing these subfragments to find some similarity and thus score. So right now, due to its importance at the search level in general terms, we are going to talk very briefly about how the BLAST algorithm works. Here what you observe is previously a sequence, of which we are going to first take a relatively smaller region. And from this region in turn, we are going to determine or find something that is known as the word, which in summary is a kind of triplet of amino acids that allows us to subdivide the sequence into even smaller fragments. And now for each of these triplets or letters, what is done in summary is to pass it through a matrix that has evolutionary foundations, which we will also review later in detail, which is called Blossom 62 substitution matrix. And the summary is that once doing the calculations with these matrices, the goal is to find, well, triplets or words that are synonymous with the triplet that we determine from our particular query sequence, to have as well as a list of candidates to search for with other proteins. As a second step, obviously now we have to apply this entire segmentation process into triplets and much more information to all the proteins to which we want to perform our query. Usually all these tools like BLAST, MMSX, or Diamond have commands that what they do is take a set of millions of protein sequences and create a specialized database to run their algorithm. And here are, for example, some of the commands that are usually used to create these databases that index each of the protein sequences that we intend to search for. Now, once it is already indexed, that is to say that this whole matter has already been done with mathematics for each of our proteins. We proceed to actually make the comparisons, but with these triplets of amino acids, in such a way that what we do is compare the protein. That interests us with respect to the millions of proteins that we have to search for, and if we find a match as marked here in red, then the algorithm detects that there is precisely this match. And it tends to look for its subsequent contiguous regions to evaluate whether this consistency continues to be preserved, which, as we can see, there are again 2 residues S, 2 residues Q, 2 residues A, until it finds one where there would no longer be a match, and then it opens this empty space that we usually call gap, which the algorithm penalizes because, in principle, it starts from the idea that, well, if I am looking for something similar, it should be similar both in number of sequences and in their identity. And in this case, it is necessary to open one so that the identity is preserved, as you can see here in the subsequent amino acids. So the fact of opening this gap, this little hole, is penalized by the algorithm. And finally, thanks to this, we can now really score which sequences are most similar to our sequence of interest. And thus, we have different confidence metrics and information that BLAST gives us, which perhaps the 3 main ones would be these following. What is sequence identity or sequence similarity, which is usually expressed as percentages, right? Like, I don't know, 20%, 30%, 64%, etc., etc. We have another metric called e-value, which in short is a probabilistic measure that tells us what the probability is that our 2 proteins look alike. Well, due to an evolutionary event from ancestor to descendant, or by mere probability, due to having compared 2 proteins. In this case, very small values such as approximately 10 to the minus 3 are usually adopted, which is equivalent to saying that our protein necessarily has to be compared 1 against 1,000 proteins, and at random we could find this possibility of them being similar, which is a relatively low probability if we look at it as there being 1 in 1,000 possibilities. Obviously, when we raise this range to perhaps values of e to the minus 5, it would mean that we are talking about that we now have 5 zeros here, and then the probability that this event has occurred purely at random is evidently quite low. So we could think that they are really 2 homologous proteins. Finally, we have another evaluation metric called bit score, which also considers the size of the database with which we are comparing our protein, and which in summary also takes positive integer values, but usually by considering precisely the number of sequences present in the databases. It can also be considered as a confidence metric that is more sensitive, and that usually it can be associated with determination of homology between 2 proteins when this metric takes values greater than 100. Here what we observe is precisely the number of homologs that we can detect by being able to do either searches at the protein level or searches at the DNA sequence level. And as you can see, when we do searches with proteins against proteins, we usually have a much larger number of homologs represented here by each of these colored bars, compared to the searches we can obtain for homologs with DNA-based sequences. They also find that even this bitcode or this quality that we have that they really are homologous, or well, that they are very possibly homologous. It is also quite considerable. In this sense, we have several candidates painted red with a bit score greater than 200 and some between 80 and 200. And in this case, we have very few candidates with a bit score of 200 and others who are of poor quality, even up to a bit score of 50. And this is actually a fairly well-known fact, which is that precisely the search for nucleotides. It is valid to be able to search for highly conserved sequences that have diverged in a fairly small number of years, compared to the vastness of the evolution of more than 4,000 million years. Regarding the search for proteins, which is much more sensitive and can find ancestors even more than 1,000 million, up to 3,000 million years old, as long as their sequences have not diverged so much. And so here a natural question to ask is, why are proteins more sensitive to be able to perform searches at the homology level with respect to DNA sequences? And the fact here, well, although there are multiple answers, perhaps the most obvious is that by having an alphabet of 20 amino acids compared to our alphabet of 5 amino acids at the DNA level, we obviously have more information that offers us greater resolution to be able to resolve the homology relationships that we want to investigate when comparing two protein sequences. And here we address the topic precisely of the relevance of what blossom substitution matrices are. These matrices are super well thought out. They are even really part of Margaret Dayhope's most relevant legacy, given that she was one of the first researchers to suggest this type of matrices of evolutionary information, such that these matrices consider the probability that one amino acid mutates to another amino acid based on the observation of millions of proteins. And here the important thing is that algorithms like BLAST incorporate precisely this molecular evolution in the form of concepts, such that they help precisely in the search for sequences based on these evolutionary relationships. So in particular, I highly recommend this review, where they really teach a lot about what BLAST means, how it operates, what things to consider, but here we are going to very briefly address how these substitution matrices work. So for example, what you can see is that here is a score that was given by reviewing millions of sequences, which is when mutating one cysteine residue against another cysteine residue. In this case, it was arbitrarily given a score of 9, and this is actually quite peculiar because when we compare how the possible substitution of an alanine residue would be scored against another alanine residue that meets the same condition of being a substitution to the same residue, we see that it now has a score that is relatively lower. We see that a cysteine to cysteine mutation is given a score of 9, while an alanine to alanine mutation is given a score of 4. So why is this happening? When we turn to see what the frequencies are, the percentages of amino acids in huge databases, such as this one called UNIPROT, which derives statistics from approximately 200 million proteins, we observe that not all amino acids are present in the same percentages in these protein sequences. There are some amino acids that are quite common, such as leucine, and there are other amino acids that are also obviously quite uncommon, such as tryptophan or cysteine. So here what we can observe is that it depends both on its frequency, precisely observed in this type of data vessels, as well as its chemical and evolutionary context. Regarding that in this case, what is a residue such as alanine? It can really be classified as an amino acid of the nonpolar aliphatic type. But this is not the only nonpolar aliphatic residue. We also have leucine, glycine, valine, etc. However, when we inspect what other residue exists similar to cysteine, well, we find that there are practically only two, which is methionine along with cytosine, because these contain sulfur atoms. So its chemistry, its evolutionary context, as well as the frequency that we observe in the databases, lead us to suggest that mutations, due to their biochemical context, are less likely to occur between amino acids from the same groups. As another example, here what we have is the probabilities, or rather what the score is, of mutating from one amino acid D to another amino acid D. In this case, we see that it has a score of 6, but what happens when we mutate to another amino acid that is quite similar, which is this one called amino acid E? As you can see, really the only difference it has is that it has a methyl plus group that makes it a little bigger. And then, observe these same negative charges indicated here in pink color in each of these peaks. So, as these two residues share a relatively similar chemical behavior, except for this extra methyl group, which makes it larger, it is given a score of 2, which in this case remains relatively similar due to its biochemical context. However, when we evaluate the score that is given when a residue D against a residue W, what we find is that it is now extremely poorly scored. In this case here, you have the amino acid W with respect to the amino acid D, and it is given a score of -4 in this sense because it is quite evident that the biochemistry of these two residues is completely different. While one has two rings with aromatic properties and a positive charge, this one, since it only has two negative charges and does not have the presence of rings or resonance. So it has properties of flexibility, of charge, that are completely different from each other, which is obviously consistent with the fact that they are being scored in a negative way. And then these types of aspects precisely allow BLAST to detect proteins that are more similar to each other based on their level, on their biochemical, frequentist, and even evolutionary information. So this algorithm was so successful given that, since it includes this type of biological bases. In this case, for example, the chemical or frequentist similarity of these mutations is penalized less, which means that this amino acid similarity is being considered, which matters quite a lot. As a second aspect, we have precisely that it greatly penalizes the fact of having to open these gaps, and not so much if the gap is so big, but having to open it. Well, it necessarily implies an act of some kind of injury or some insertion of a new amino acid, which evolutionarily, well, let's say that it is also less likely to occur. So this is penalized even more and even well. What local alignments are really, what they help us with is to model even better what this modular nature of protein domains is. Since, well, we can have a protein, well, quite large, composed of multiple modules, and that by doing a more segmented search, more of small fragments instead of a global search, it allows us precisely to detect with this degree of sensitivity the modular nature of the domains. What we observe here is now how fast they are. I told you, these two algorithms called BLAST or MMSX. Regarding what is the algorithm that has historically been used called BLAST. Here we have precisely BLAST as a reference, and here what is also being able to classify two homologous proteins that evidently share ancestor-descendant relationships. And what we observe is that these two programs have different parameters that one can play with to determine the sensitivity with which we want to detect whether or not there is a probability that our proteins are homologous, in such a way that you run the algorithm and indicate the level of sensitivity using words like fast, sensitive, very sensitive, or ultra-sensitive. Or in the case of the MMSX algorithm, it has a parameter called S, which takes values from 1 to 7.5, with 7.5 being the most sensitive version it has. And what we observed is that, when we use the default features that these programs include, it turns out that, for example, Diamond in version 207 runs up to approximately 8,000 times faster than BLAST. No more than if they can notice that the sensitivity that BLAST offers us is being lost. However, when we run each of these algorithms in their most sensitive versions, it still turns out that MMSX runs approximately 13 times faster than BLAST, offering almost or rather practically the same sensitivity, and that Diamond, even when we put it in its ultra-sensitive variant, runs up to 82 times faster than BLAST, which is obviously super practical when we are searching in databases that have millions of proteins in that sense, because really the suggestion is to use one of these two techniques that are much more optimized. Regarding what BLAST was in its first steps, and that even this has been tried to improve, these values have already been improved a lot, so that they are encouraged to adopt these tools such as MSX or Diamond, and that they have even been adapted to be able to perform calculations at the GPU level, which here we can find some comparisons in what is the construction of the multiple element of sequences, or what is the prediction of a structure that obviously has this step, but has to run what is the AlphaFold algorithm to predict the sequence. And what we found is that by being able to adapt the MMSX algorithm now to computations performed from GPUs, there is a reduction of up to 177 times in the time necessary to build an alignment. And in this case, when we want to predict a protein structure using the AlphaFold algorithm, you can reduce the speed up to 22 times using this MMSX variable on GPU. So this will even scale the capabilities we have to search for proteins already to billions of proteins perhaps. And perhaps now there are even tools that no longer only consider what an amino acid alphabet is, as we can see here, which we are already quite associated with. It is something that is relatively common. But now they also consider a structural alphabet, which is also defined just like the amino acid sequence alphabet. Now we have a structural alphabet that also has 20 symbols, and obviously by now having structural information encoded in the form of 20 symbols, we can now combine this sensitivity that gives us the power to compare protein structures with respect to comparing sequences, but do it at the sequence level and obviously do it much faster. And we are obviously going to cover this in the class on protein engineering based on artificial intelligence, where we talk in more detail about this type of algorithms. Now it is relevant to recognize something that I have been talking about, which is this concept of homology, that what it really indicates is whether it exists, or if there is a probability that it exists. An ancestor-descendant relationship between two proteins. So here the question I ask you based on what we now recognize of protein similarity is whether these two proteins, the class A beta-lactamase called TEM1, which has 286 amino acids, if this protein is homologous to the green fluorescent protein which has 236 amino acids. At first, when observing their structures, we would think that they have nothing to do with each other. While one is practically made up of many alpha helices and a few beta folds here, this one practically in its entirety has many beta sheets, and here there are only a few small regions of alpha helices. So, based on the structural observations we just made, we could hypothesize that no, they are not homologous, they do not share a common ancestor, and they do not have a truly shared history. And now I present to you a new protein, a protein called penicillin-binding protein. Now the question is this protein homologous to the class A beta-lactamase TEM1? It has approximately 30 more residues, but now we see that their shapes are relatively very similar. Then we are going to carry out an overlap of their structures. And this is what we see. They share a sequence identity of approximately 4%, and their RMSD is 5.62 Armstrongs in the case of comparing GFP against TEM1, which is telling us that these proteins have nothing to do with each other in structural terms, and that we could think that they are not homologous. Now when we compare the PPP protein with respect to the TEM protein, we find that they have a sequence similarity of approximately 21%, and that their RMCD is 22.5 Armstrong distance, which here is really reflected in structures that are relatively consistent despite the fact that there are certain regions where perhaps there is greater structural diversity. Then we could think that, well, indeed, perhaps they are homologous due to their structural similarity. Now, what happens if we compare a class A beta-lactamase TEM1 versus a class C beta-lactamase called S1? These are two homologous sequences. So when aligning the structures and calculating their TEM score, what we find is an RMSD value of 3.66, the TEM score value is 0.7, and the identity or sequence similarity that exists between these two is 12%. Here is the value of the TEM score, which, remembering values greater than 0.5, are suggestive that it is the same folding at a general level. We can think that they are homologous proteins, despite the fact that, as we recognize here in these structures, the blue protein, which is the class C beta-lactamase, has a much more considerable structural diversity compared to the class A beta-lactamase, TEM1, which is the one presented in orange. We see that class C beta-lactamase is relatively larger, has more alpha helix regions, but shares certain regions in common with class A beta-lactamase. And this is quite interesting because how can it be possible that the sequence identity becomes higher, as we saw in the previous case, between a class A beta-lactamase with a PVP, and now when we compare two beta-lactamases, it turns out that they have a lower sequence identity. And this is evident when we look at what the evolution of this type of enzymes was. In general, beta-lactamases are thought to have evolved from what are known as precisely PVP. However, the types of beta-lactamases actually derive from different ancestors of these PVP, such that, for example, what beta-lactamases are class A derived or are thought to have derived from this type of proteins called class PVPA, and in turn from these other low molecular weight PVP. And in this case, class C beta-lactamases were derived from another type of protein that is also evolutionary related, called AMPH, and in turn, these were also derived from another type of PVPS. So even though they have names that are relatively similar, which is class A or class C beta-lactamases, when we look at their evolutionary history again, it makes a little more sense that, well, they become more dissimilar to each other when we compare them with a PVP, which would be their direct ancestor in that sense. And this is something relatively interesting because this evolutionary distance is not really reflected in terms of the percentage of identity that two proteins share, such that when we find a similarity of between 70 to 80%, we could think that the scale of divergence we're talking about, therefore, covers a few 100 million years. But when we find divergences at the sequence level that are already spoken of between 20 to 30%, we are talking about relationships that led many billions of years ago in evolution, such that we could think of scales of approximately 10, 3,000, 4,000 million years that these relationships would have diverged, and that even with current algorithms, we can detect these probabilities of them being homologous. Here, a relevant concept that must be recognized is what is called the twilight zone, the zone like the sunset, which is a region where it is no longer really certain. There is no probability, there is no certainty that we can determine homology between two sequences, because the data simply no longer gives us, it is not really reliable in this sense. What does this mean here? What we observe is a comparison between pairs of protein structures, where we take 10 random domains from a database called SCOPE, and which are proteins of approximately 20 to 250 amino acids, and we compare them. Pairwise metrics are already made of sequence similarity as well as structural similarity, seen as the RMCD. And what we observe is that the sequence identity, seen here in a range of 0 to 10, and here the structural similarity of a range of 0 to 7 RMCD, which is measured in Armstrongs, we observe that when there are 2 Armstrongs of difference between pairs of proteins, it is therefore reflected in a sequence similarity that ranges from 40% to 100%. But when we talk about a shared sequence similarity of approximately 30% or more highly 25% sequence identity, the structural diversity that we can find explodes in an incredible way, such that then there will be some pairs that actually do share this structural similarity, such as this whole box here, that actually share a similarity of 2 Armstrongs or less. But the most likely thing is to find cases where this RMCD explodes up to values of 7 or 6, which would be indicative that they do not have a structural similarity. So this twilight zone is precisely this range, where it really is no longer like us. Really so consistent that this sequence similarity is reflected in terms of the three-dimensional structure of the protein. Now seen in probabilistic terms, we can also evaluate this twilight zone in the number of residues that have managed to be aligned, which is precisely what we observe on the x-axis. And on the y-axis, we observe how similar two sequences are, in such a way that precisely the number of residues that we are considering also matters when evaluating whether two proteins are homologous to each other. In this sense, when we compare sequences of approximately 100 residues and their sequence identity is 80, we could have some certainty that indeed this similarity at the sequence level. It would also translate into similarity at a structure level. However, when we now also compare proteins that are small, such as approximately 50 residues, and these 50 residues share only 25% sequence similarity, here in this area, we are no longer likely to think that they were or were not merely random relationships. Because when we compare two completely random proteins, they usually show at least 5% sequence similarity. So when you are comparing either protein sequences or protein structures, you have to consider these ranges of the twilight zone so that when we are comparing sequences that happen to share, they share less than 25% sequence similarity, we will necessarily have to look at how their protein structures are reflected, since then in this case we no longer have a high probability that these relationships become truly homologous. And also consider the number of residues obtained from the alignment to evaluate the amount of information that the alignment offers us. And consequently, the probability that these alignments have to offer us this determination of homology or non-homology, which is a binary attribute. One is not more or less homologous, but rather one is or one is not homologous. So here's a kind of summary of the type of evolutionary signal that we can use to be able to infer certain types of relationships, where what we find is precisely on the y-axis. What would be this evolutionary signal, or the phylogenetic signal regarding the depth of the relationships, or properly the divergence? So then, really when we want to discriminate relationships that have occurred in a relatively recent time in evolutionary terms, we can usually investigate what are the changes of a single nucleotide in DNA chains. When we talk about relationships that are relatively superficial, that span several 100 million years or perhaps even 1 billion years, it is more convenient to be able to use gene sequences, protein sequences, or even the structure of the protein itself. But only in the case that we want to investigate homology relationships that are quite deep. Until now it seems that only the information derived from the structures of the proteins is capable of investigating these types of relationships, since as we already saw, the structure is preserved up to 3 or up to 10 times more with respect to the divergence that occurs in the proteins, and that only in extreme cases of evolutionary divergence. Well, now we have to use multiple analyses at the sequence level, at the structure level, at the level of structural submotifs to be able to establish whether two proteins that we suspect are homologous really are. That is to say, they occur, or rather we have a catalog of tools either at the sequence or structure level to be able to determine with greater or lesser resolution the possible homology relationships that may be occurring between two proteins. So as a summary of all this, we have that proteins have physicochemical bases, which are their molecular interactions, their geometric limitations, which in turn really constitute these units of secondary structure that are stabilized by these molecular interactions, and that due to their nature, we can also classify amino acids in general terms as hydrophobic, hydrophilic, among other properties. As point number 2, we have the recognition of what its folding patterns are at a 3-dimensional level, which we know as folding. As a third, we have that if we have a gene, and this gene can really contain multiple domains, which is the unit that is recognized as one that can fold by itself to even perform its function independently of the other, despite the fact that they are encoded in the same gene. They are recognized as a domain and are usually associated with a single fold, although with some nuances that we will investigate later. As a fourth point, we have that there is homology at a structural level, when we observe that although thousands of years of evolution have passed between what is an archaea and an algae, when we investigate the similarity that occurs at the level of their structures, we find that there are quite consistent patterns despite the billions of years. And we also talked about the fact that there is a homology that can also be determined now at the sequence level, where perhaps it is more relevant to turn to see precisely if our comparisons fall in these safe regions of homology determination, or if we have to be more careful because they fall in this region where we are no longer certain that these relationships are homologous or not, because they pass precisely through this interval known as the twilight zone. This is all we have to consider as a whole to look at how the current classification schemes work. In fact, perhaps the two most famous are precisely the scope and the CAD, which have different categories in their cases. Here, for example, there is an equivalence of the categories that are used between each of these two classification systems, the highest category they have in these cases. It has the same name, only in this case in the scope classification, 4 classes of proteins are recognized, while in the CAT classification, 3 classes of proteins are recognized. Because the authors decided, given their analysis, their particular considerations to determine this number of classes. But if you remember, they're pretty consistent with what Cloche's suggestions were about them being all beta, all alpha or alpha-beta combinations, or those interleaved versions. In the case of CAD, it has a category that does not have a direct equivalent in the scope classification, which is called architecture, which considers what the orientation of its secondary structures is. Now we have a third category that is equivalent in this sense in both databases, only in the scope database. In the scope classification, it is known as folding, and in the CAD classification, it is known as secondary topology. But in plain terms, they share similarity, given that they precisely consider this connectivity between elements of secondary structure. And finally, we now have a fourth category that is also equivalent in these systems, where in scope it is called superfamily, and in CAT it is called homology, that now recovers information at the level of what the sequence of the protein is to be called superfamily, and that in this case scope. It has one more category, which has no direct equivalent in the CAD category, which is called family, which are precisely protein sequences that share at least 30% sequence similarity. There is also another database, another classification system called ICODE, which is also often used quite a bit, but it was also evidently created based on the considerations made by its authors. And when we compare how consistent the proteins classified along these categories are between these 3 main classifications, we find that generally speaking, in what it would be between the ECODE and the CAD, there is actually quite considerable structural consistency when we investigate what the proteins deposited in the PDB protein database are. And when we compare the ECODE and the scope, which is a European variant of this, which is the scope, they also find relatively consistent relationships, at least at the level of determining which regions mark the end or beginning of the domains. So really the choice of which database to use may depend on precisely whether or not you agree with the guidelines on which each of these classifications are built, as well as perhaps some tool that you are using that perhaps offers more information associated with a database. Then perhaps the adoption of this database is more advisable because it contains a greater degree of information. It will depend specifically on the question you want to answer, as well as the information you have. To be able to make the choice of one of these databases, which include either manual curation or curation based on automatic alignments. Here the idea is that in general terms, these classifications of protein structures have general categories such as class, fold, superfamily, family, protein, and even species level, right? That is, we also generate these types of ranges that we saw when generating this taxonomic hierarchy, such as the phylum, domain, class, order, family, genus, and species that we as biologists use, only now at the level of macromolecules and, in this case, specifically at the level of proteins. However, we are not really clear, or there is a fairly well-defined consensus on what we are referring to when we talk about a fold, or what we are referring to when we talk about a superfamily. And this is quite clear in this particular table, where there are different authors who ask the question. So, based on our calculations that we can do, and with the information that we have in different periods of years, as you can see some carried out from 1992 to 2009, we find that there are those who suggest that there may be less than 1,000 faults practically in our reality. And there are some who suggest that there may even be 10,000 F. In another comparison, there are those who suggest that there could only be about 1,500 superfamilies of proteins, and there are others who indicate that there could even be up to 50,000 superfamilies of proteins. That is, really, although these systems help us communicate with the object with which we are working, or the protein with which we are working. Well, there really is no consensus on being able to define what we are talking about when we are talking about a folding, or what we are talking about when we are talking about a superfamily, and consequently, when we really work we do it from the premises that we take when using these databases, which really complicates a bit when we want to do more general analyses, and we have to accept that. When we communicate our results, they derive in part from the choice of one of these bases or structural classifications. Here we can see an example of how some proteins are organized in the CAD. For example, we have what their classes are, which in this case would be the majority alpha, the majority beta, or combinations of alpha-beta, or very few structured regions. So in this case, let's say that we are interested in what proteins are that have combinations between alpha and beta. And within them, we now have a second index. In this case, it is index number 3. Now we have an index which is 40, which is now called a 3-layer sandwich in particular. And now, within this architecture team that is 3 layers, we now have different topologies such as, now indicated with a new digit, which would be 53550, where we now find slightly more refined names, such as L9 ribosomal protein, fructose permase, or rosmin. And even if we now turn to look at precisely this particular topology, we find that within this specific group, there are 278 superfamilies, which look more or less like this, which is reflected as a new digit in this nomenclature of 4 digits separated by a dot, implemented by what is the CAD system. So here you have different superfamilies where what you observe is precisely the regions that they have in common with each other, and that there are also regions in the structure of the protein that are considerably different and that are not marked in red. So you find that there is a central part that is conserved, and that superfamilies are precisely characterized by this diversity at a structural level. If you are wondering where I can access this type of information from these structure classification systems, it is relatively easy. Obviously, each of their web pages contains all the information, but they are also stored in what are the annotations on the InterPro page, which is a sequence database. Or what is the page of the protein structure database, that is the protein data bank. In this case, when we search for this protein using this index, we see that the PDB displays a lot of information and organizes it in the form of tabs. And if we go to the Sequence tab, we will find this annotation that provides us with multiple annotations that exist in each of its residues. and that in this case we could see, for example, what is the degree of hydrophily along the sequence, the disorder estimated to be the same along the sequence, or if there are, for example, some residues that have been previously detected. As important, because perhaps they are acting precisely as a binding site for some ligand. But when we look again, here in this orange part, there is also information regarding its annotation at the domain level by these databases. And here we can more easily find what type of protein it corresponds to. If you want to learn more about how to interpret this type of data, you can check out the seminar on YouTube with a lot more information. And even in what is the AlphaFold database. When investigating these maps, here we are also shown with these colored regions to what type of domain each of our proteins that has been predicted with AlphaFold would correspond to. So now we move on to the third topic of this class in total, which is to investigate what the behavior that we have observed in recent times of this universe of proteins is like. Here what we will address are practically new perspectives that have been generated, as we have obtained more information both at the level of protein sequences and protein structures. The first point being the fact that really the distribution of proteins in this space, in this universe of proteins, is really skewed such that there are even only a few folds that really recapitulate the vast majority of proteins. As a second point, we also have that there is obviously more information, and evidently from it we can make new observations, such that we have begun to find new relationships of homology between foldings that we previously thought had no evolutionary relationship with each other. And finally, we will address new paradigms that are occurring within what is structural biology, where we are really beginning to recognize that what is the structure of the protein is much more flexible than what we had previously thought or observed. And here, as a little curious fact, there is a researcher with quite interesting illustrative abilities called Irina Vesnova, and who has an art gallery that usually applies new designs every October where you can review these types of illustrations related to what the structures of proteins are. So, as we saw in this first point, there is a distribution that is quite skewed with respect to protein folding. And this is an observation made even since 1994 in this publication by two of the greatest researchers in protein biology, which would be Christine Orengo along with Janet Thornton, as well as David Jones, who in this case, when observing, with the information that was available at that time, which was relatively, well, something sufficient, but evidently not as much as what we currently had, they found that even just 9 volts are those that represent up to more than 30% of all the structures that had been deposited in the PDB at that time. And on the right what you see is precisely an image of each of these 9 structures, of these 9 volts, and this is quite interesting because now we wonder what conditions led to the success of these volts be so overcrowded in terms of the protein sequences. And the common characteristics shared by this type of folding, which due to its success have been called superfolds, is that they contain simple topologies, they have a high content of secondary structure, their kinetics during the folding process are also considered relatively simple or easy to conceive. They are also structures that are usually quite thermostable, and that evidently thanks to this whole set of properties, have presented enormous functional diversity with respect to the catalytic activities that enzymes containing this type of folding can perform. These are observations that are actually quite interesting, and that were even replicated by another group that also adopted a little more information for those moments, such that they reached relatively similar conclusions, where we observe that they even suggest that 40% of all domains can be covered with just 16 types of faults, which then recapitulates this idea that there are certain faults, which we could call super faults, that are highly populated. And here they also make other quite interesting observations regarding the nature of these super faults, so that, well, perhaps this great stability that they share with each other could be like a reflection of an economy, of what is the construction of their multiple modules. And another observation they make is that curiously these superfolds also share, or rather the domains that are present within these superfolds share certain structural signatures that are these tiny fragments of structures that we previously reviewed, that possibly represent what we can say as nucleation centers that, during the folding process, seem to promote precisely a much more harmonious folding, much simpler to achieve compared to other folding processes that can be considerably more difficult, and that even all this diversity that occurs during the folding process is addressed within a complete class in what would be the functional activity of proteins. When we see how current this data is that only a handful of folds recapitulate the vast majority of proteins, we observe that even with observations made in 2023, quite similar patterns are repeated, which in this case approximately 5% of what are the superfamilies cataloged within the CAD classification represent approximately almost 60% of all the domains that this database has. Here, as you can see in this PAI graph, what is observed is precisely that the 100 most abundant superfamilies within this protein classification represent 54% of the domains. And if you are curious to investigate what forms these types of domains present, here you can evaluate this other analysis, where they also present what the PDBIDs would be to be able to access them at a structural level, and verify what properties make them so abundant, including comparing the abundance between multiple databases, such as the COP, the CAD, or the dictionary that also exists from another scheme called DALI. This is also reflected at the level of protein sequences, so that even the majority of families are represented by practically a single member. This observation has been made in multiple studies, but perhaps this is one of the most up-to-date, where what they do is take a set of approximately 1 billion genes. Subsequently, we group them at 30% sequence identity at the amino acid level, and as a result, we obtain what would be 32 million of what the authors call unigenes. And then with them, we're going to see how many families they are associated with. And what we observe is that even 80% of the protein families are barely represented by 1 or 2 members, which can be seen here in these 2 Qs, which are practically only represented by too few members, which also has multiple observations, in the sense that this information also derives from what is known as metagenomic analyses. And perhaps during this translation process of these sequences, they are not really experimentally proven to exist, but we can have good approximations that they do, because perhaps they would not be translated correctly, such that then they would really be spurious sequences. But it is also likely that even these types of genes, which are quite few, but represent millions in this sense of unigenes, they can be like what is considered de novo genes or de novo proteins, which we say are inventions that come to be but relatively spurious, that their time window is quite short, so that in this type of genes or proteins, natural selection really operates differentially with respect to genes that are, well, let's say that those of the electron transport chain that are vital for many forms of life are quite well known, in such a way that this type of genes that are represented by very few members are not usually associated with an adaptive value in general based on the relationships and phylogenetic analyses that were carried out in this study. So perhaps they could be like this engine of diversification that evolution finds for, to pave the way in what is this phenomenon called life, and that well in this sea of solutions, well, some proteins will perhaps be successful, but well, perhaps the vast majority, as perhaps suggested here in this scheme, that 80% consists only of 1 or 2 members. Well, that would be the case also as a third observation. We have that, well, thanks to the fact that we now have more information already at that time. There were approximately 200,000 structures of proteins deposited in the protein database that is the PDB. We found that we can evidently generate new observations and link homology relationships between proteins that we previously did not think had any evolutionary relationship. In this case, it was even demonstrated in two of these superfolds, which in this case is called the beta-alpha 8 barrel or the flavodoxin-like fold, which, by finding what would be a kind of intermediary, homology relationships could be determined. So what we observe on this side are some little circles where, let's say in simple terms, we draw a line as long as our algorithms and the information with which we analyze become capable of detecting homology. So, at least among the members of this superfamily, it is relatively easier to detect these types of evolutionary relationships. However, when we analyze the members within this other superfamily, we find that it is a little more complicated to determine homology relationships. But it turns out that here the authors found what would be a kind of intermediate stage that shares certain properties that are common to both types of superfolds, and with which they can precisely detect that there are homology relationships, which invites us or suggests that in reality these two superfolds, since they would share a common ancestor that is reflected by these small structural signatures highlighted here in different colors in each of these folds. An observation that, as we saw previously, these subsignatures also have an evolutionary value, given that they are usually found in multiple foldings that we think may not have an evolutionary relationship, but possibly through detailed analyses such as those found here, we could observe that these types of relationships do exist. This is evidently addressed a little more in what the class of protein evolution is, given that all this theory behind what structural signatures are is also thought of as an important link during the first steps that were the evolution of proteins. Another point that is also relevant to consider is that what structural similarity really is does not necessarily translate directly to homology relationships. And this may be because similar structures can even arise independently through convergent evolution events. Here, one of the perhaps most surprising cases is one of the most abundant foldings that we know of, which are called Rossmann. And where in this particular research, the authors are in charge of defining what would be the minimum structural signature that we can associate with this type of Rossmann folds. So they define this structural motif, and then now they search for it in the entire database of protein structures that we had at that time, which is the PDB, and they find that this structural motif, even though we do not consider it as a FOD in itself, but as a structural signature, it is present in 20% of all structures known at this time. And this, at least the authors suggest, is a convergence event, which could have occurred for, among many reasons, these two that would be the essential ones. One is the fact that this motif is usually associated with high thermodynamic stability, which is quite desirable in terms of the folding of any protein. And also to the fact that during these first steps that occurred during the evolution of proteins in these ancestral proteins, because of physicochemical evolutionary conditions, both of the environment in which they developed and of the genetic code itself, as well as the amino acid code, well, there were quite limited solutions regarding what could be resolved with what was available at that time, right? Something that, being abundant at that time, perhaps permeated quite a bit throughout the process of protein diversification throughout evolution. As another example, we also have here a research that is relatively, or rather quite recent, where what they do is a review of multiple cases where what is known as evolutionary convergence was presented in terms of the active sites of enzymes, which in this case present a compilation of approximately 34 cases where this phenomenon occurs. And here what we see are two proteins that have completely different foldings, as you can see, but that are capable of carrying out this biochemical reaction known as carbonate hydratases in such a way that, although they have completely different foldings, their catalytic mechanism is relatively similar and is also based on these residues. they share in common, and that even when we overlap their three-dimensional structure, the arrangement of these catalytic sites is preserved in a relatively similar way, which are, let's say, one of the few solutions that life found to be able to carry out these types of biochemical reactions. So now what we observe as a whole is that in reality convergence events, although they can be considered rare, they do have an importance, at least in evolutionary terms, that perhaps we had not recognized until relatively recent times. So now we come to one of the questions that causes me a lot of curiosity when observing these two types of folding. One protein is called Ga98, and another protein is called GB98. So, when looking at their structures, one of the natural questions we might ask ourselves is, are these two proteins false homologs? And perhaps the most natural answer would be to think no, because one is composed mainly of pure alpha helices, while the other only has one alpha helix and a predominance of folded betas. And it turns out that they are homologous at the sequence level, as you can see, because their entire sequence is practically shared in its entirety, except for a single residue, which is indicated in cyan. In the case of Ga98, it is a leucine residue, while in GB98 it is a tyrosine residue, and since this mutation was carried out, its results at a structural level are completely different, such that the folding changed completely. This type of behavior that can be exhibited by a few proteins is known as switch faults. In this case, some analyses have suggested that even the proteins present in the PDB can find cases of switch faults in between approximately 1 to 4% of all the proteins that have been deposited in this database. This particular case is not the only one. There are multiple other cases of switch faults recorded where a single handful of mutations can actually create faults that are practically completely new, or make folds that were previously simple, now more elaborate in structural terms. Here you can see at least these three examples. Precisely the one in the center is the one we just reviewed on the previous slide. But in this case, we now have a protein known as the ARC repressor, which evidently only has two mutations. Now it changes its configuration, and now has two folded betas that configure an interaction that gives it completely different properties from its previous stage, which was one with alpha helices. And here we have other configurations of subfragments in a protein, in this case one called NW1, and obviously with two mutations we can change its behavior in structural terms, despite the fact that there are only a handful of very few mutations, and that perhaps it would not be a behavior that we could expect, since they are quite few. Here what we see is really how this change process occurs at the conformational level of secondary structure, in such a way that precisely these switch-fold proteins can completely change their secondary structure regions, and that they are really associated with what would be their function within the cells. Here what we see is that in the case of this protein, when it is in its alpha helix configuration, it cannot bind to its protein, which would be the target or its partner with which it usually interacts. But when you adopt what would be the beta sheet configuration, now you can join. And so this conformational diversity that occurs in this type of switch folds is now also seen associated with physiological processes within the cell. So we could think, perhaps hypothesize, which therefore have some evolutionary value in this sense. Even so, these conformational changes at the level of secondary structure can be precisely triggered by multiple biological processes. Among them, we have mechanisms such as what would be alternative splicing. We also have mechanisms that can even arise only due to mutations of a single nucleotide, which are these polymorphisms. We still have post-translational modifications that, for example, adding a phosphorus atom can completely change the behavior of our protein. Or even also at the level of the ribosome. How is this ribosome at what speed? How fast is it carrying out its translating activity? Well, it can translate proteins that are completely disordered, or it can translate proteins that are well folded. There are multiple biological mechanisms with which this structural diversity is usually associated, which evidently also translates into functions for the cell. That even, for example, Here what we observe are cases where a protein of this type, switchfold, is found in proportions 1-1 or 50% 50%, whatever you want to say, under physiological conditions, as is this case of a protein called PIMA, which in different stages is also capable of contributing to the function of the cell itself. Even we have been able to take advantage of this property, that some faults can interconvert their secondary structure regions based on multiple properties, such that even these switch faults have been designed using what would be temperature information, which by breaking some links that would be. It turns out that a conformation that is adopted at 5 degrees now changes completely when the protein is presented in a 30°C condition. This has really provoked us. It has encouraged us to rethink what we conceived regarding the structural space of proteins. So before what we thought was that there really was a quite discrete space, where each of these regions are probabilistic regions. That is, here we have a greater density of proteins that can be associated with a folding of the alpha plus beta type, and that these are completely separated from what would be foldings of the beta-only type, or the alpha-only type, or the alpha type and interspersed betas. But it is thanks to this type of full switch mechanisms that perhaps now we can navigate in a more fluid way in this structural space. Regarding what we had previously thought was the behavior of these proteins, that although we did recognize a high dynamism in what these proteins are in cellular terms, perhaps we did not think in structural terms that their secondary structure regions could change as much as we are now beginning to investigate. And these are relatively recent topics that have had a boom since 2015, mainly by a researcher named Lauren Porter. And it is expected that this type of information will even develop further, and that it will allow us to recognize that the structural space of proteins is evidently much more fluid than we thought. So with this, we reach the conclusions of this particular class, which serve in part as a recapitulation of this entire exercise. So as a first conclusion, we have that what is the backbone of a protein? It would represent what is its central body, its most relevant structural part, while the amino acids in particular, specifically their side chains, define their chemical behavior. As a second conclusion, we have that we as people, since we organize or create these categories of saying, this is the primary, secondary, tertiary, or quaternary structure of proteins, just as we create structural units that we refer to as folds or domains to have a better understanding of what the protein structure is, and which we now see is much more diverse than perhaps we had thought. As a third point, we have that the structures are more conserved than the sequences and however, both representations of what the protein is offer different levels of evolutionary signal to be able to resolve certain analyses. Now we know that in order to be able to attend to fairly deep evolutionary relationships throughout evolution, perhaps it would be more interesting to explore precisely the information derived from the structures, while in other conditions, what genes are at the nucleotide levels or the protein sequence would be better applied. As a fourth point, we have a quote that we saw from other of the articles that we investigated where there really is no definition, a consensus of what really represents structural similarity. We already addressed this precisely in that series of slides where each algorithm has a set of premises, a set of algorithms and thresholds to be able to say that two proteins are similar to each other at a structural level. As a fifth point, we have that the universe of proteins is not really equally distributed. There are some proteins that, due to their historical conditions, have been more successful throughout evolution and are overpopulated in this sense, perhaps due to their great thermodynamic stability that has given them great diversity, also in functional terms. And finally, we have the sixth point, which is that this structural space, this false space, it is really much more fluid than we had previously thought, which invites us to detect new relationships of homology, as we have also obtained more and more information at the structure level, as well as at the protein sequence level. So with this we end the class, and that would be all for the moment. Thanks so much.